Good evening. Welcome to the webinar VOYO, Voice of Youth for Oceans. Happy Oceans Day for all of you. And this webinar is a platform where the youngsters can present their work on biodiversity, taxonomy, and related fields. And why youth? The answer is very simple, because in the post-COVID era, the research scenario is going to change, primarily in terms of the availability of research money and the difficulties in fieldwork, and of course the difficulties in international travel and attending workshops abroad. And these are going to be the challenges in the immediate future. And how are we going as a group to tackle these issues? And also how better within India we can increase the possibilities for collaboration. And also with the limited resources, how better we can efficiently do the research. And also how better publications can be brought out with the limited resources. And these are the points probably we can discuss. And throughout the world, as you are aware, the youth, the youngsters are making up a large number of smaller groups, and they work more efficiently through collaboration as well as networking. And networking is one of the efficient way in which you can overcome some of the hurdles, not all. But some of the pertinent issues are still there in India. For example, those who have those very good and efficient and committed PhD holders, they are searching for job. Particularly those who have completed work in biodiversity and taxonomy. All these issues are there. So all these issues can come up during the discussions after their presentations. And this is a random pick actually of the youngsters working in India from in different laboratories. And some of the issues are very common as you work as a youngster in a laboratory, and some of the issues are very common. And some of the issues definitely we can overcome if we have a larger group and if we have better networking. And youngsters are very important, especially when you look at the future of the biodiversity in the country. And after the initial work of the Britishers and especially the faunal volumes, you can definitely see actually a lethargy as far as Indian taxonomy is concerned. And in the recent past, actually, the things are changing and more integrative taxonomy is coming up. And there are better publications with the help of collaborations with experts. And some of the positive changes are happening now. And this is a time in which even the youngsters have to think with a broader perspective, rather than thinking about publishing somewhere something. It's actually a time to bring out the quality of the publication so that the youngsters in India will be heard across the world. So this is actually a time. And with these introductory remarks, I now request Dr. Baban Ingole to express your views.
Good evening to everybody. Is it audible, Dr. Biju Kumar?
Yeah.
Good evening to everybody and my co-panelist. Namaskar to all of you. On this occasion of the World Ocean Day, I wish all of you, my co-panelists as well as the participant and all who are viewing this webinar, compliment and best wishes for the World Ocean Day. Dr. Biju Kumar, thank you very much for organizing this. I thought it is very appropriate time you have taken a lead, especially with respect to the youth. There are a lot of issues related to the taxonomy in country. We'll be discussing in time, and certainly youth needs to be a central point of our discussion, so that they are the one who will take a lead and come out with the interesting data in future. We all will be there for guiding them. There are many of expert in the country. We have to trap them, we have to involve them, so that the best will come out of it. Thank you very much once again for giving this opportunity.
Thank you, Baban sir, and over to you, Sanjay.
Okay. Good evening, everyone. Thanks a lot, Biju, for inviting me. And basically organizing this. I think it's a wonderful opportunity for everyone to share their experiences, thoughts, and what we need to do in the coming days. It's been a challenging time, and I think this format has really taken off. And as we see more and more people doing this, we find this most probably will become the norm. And once we get these initial experiments sort of well sorted, I think then we can have more such webinars, and which meetings and symposiums. So I think it's a great opportunity, and thanks a lot again. And I look forward to listening to all the presenters. Plus, as Dr. Biju Kumar said earlier, this is an opportunity to also have a compilation of all the presentations, plus complement it with people's thoughts and recommendations for what needs to be done to be published in ZooSprint. So I look forward to listening to all of you presenters, plus hearing from everybody here who's interested in getting their voice in. Thank you again.
Thank you. Now, Dr. Rajiv Rakavan.
Yeah, thank you, Dr. Biju, for organizing this initiative. I think it's one of the first of its kind, because not often do early career researchers get this opportunity to talk about their work, their issues. It's not just about their work, like you said. It's mostly about their future career prospects in the wake of all this unprecedented challenges that the whole world is facing. So I think it's a great opportunity, and I would consider myself as an odd man out here, because I primarily work on freshwater ecosystems. The thing is, mostly marine and freshwater have been managed and conserved rather in a very exclusive manner, without much collaborations and all that. But when you look at these ecosystems, ultimately both are connected, and I think there should be a need to have more synergies, more opportunities for cross ecosystem or cross realm collaborations and so on. So I think it's going to be both a learning experience for me today, hearing the problems, the opportunities that most of these young researchers face. So thank you.
Thank you. Dr. Deepak Samuel.
Good evening, everyone. And it's a pleasure and privilege to be part of this exercise. And first of all, wish you all a happy Ocean Day. And I profusely thank Dr. Biju for making an arrangement like this, where the youngsters can voice out, their research interests and what their aspirations are. And definitely, you did have aspirations, but these 3 months and the days ahead, we know that the difficulty, because in our own lab we see, especially taxonomists work on microtaxonomy, cannot access the labs. And if you work in biotech, then the kind of chemicals or other kind of post quarantine arrangements have been done in institutions. So there's a big challenge. But as my other senior panelists have told, it's a great opportunity to listen, because you guys are just you've seen your work, many of your work, and the kind of potential you have. So I'm just sitting fingers crossed and just going to enjoy the session. Thank you so much.
Thank you. Thank you, Dr. Deepak. And now, Dr. Dipani.
Okay, sadly, hello everyone, good evening, happy Oceans Day. I don't know, I hope y'all can hear me. I cannot put my video on.
Yeah, fine.
Well, everybody, all my senior panelists have already said some of the most important and inspiring things to all the young researchers out there. I would really like to say thank you to all the young researchers for holding on. I know that our field of work is not very, what they say, not in the job market, but we still do what we do. So thank you for that. I was part of an international conference this year, and I realized the best part of online conferences and online meetings, other than, of course, our fossil emissions are reduced and all that, is the fact that everybody can be there on the same platform. There is no hierarchy. There is no intimidation. Students are not scared of speaking up because they're in their comfort zone talking to the rest of us. So I think it's a great thing. Irrespective of COVID, we need to continue these interactions. And I am waiting to get back to field. So don't worry, all this is going to pass very soon, and we are going to be out there very soon. And I hope to enjoy the session. Thank you, sir.
We also hope that, because without field, there is no taxonomy and biodiversity document.
Yeah.
Okay, thank you, Deepani. And we'll straight away start with Dr. Prakash Sanjeevi's presentation. Dr. Prakash Sanjeevi is scientist in the Center for Climate Change Studies, Satyabhama Institute of Science and Technology. And his specific interest of work definitely is including taxonomy, reproductive biology, and evolution. And his model organisms are Canadian shrimps, and he's specifically interested in the association of Canadian shrimps with the coral reef organisms. Prakash.
Yes. So shall I start sharing the screen?
Yes.
Yeah. Is it visible?
Yeah. Go ahead.
Thank you very much, sir, for the wonderful opportunity on the occasion of World Oceans Day. I'm mostly interested in understanding the cryptic species diversity in the Canadian shrimps based on the integrative taxonomic approach. So why I selected this shrimp is because it's been almost 3,400 species worldwide, and it's widely available in the coral reef areas of Indo-Pacific as well as the Atlantic regions, and it has a remarkable diversity in case of morphology or in case of ecology or in terms of biology. And it helps to maintain the ecological balance. In this case, it has been very less studied, and these shrimps are mostly colorful, so they have highly exploited. It has been highly exploited for the marine aquarium trade. So how we will do it in the sense, first we will be understanding what are the mechanisms that are underlying the process of speciation, and then we will use various parameters to understand whether in terms of morphological taxonomy, or we will include molecular taxonomy and other parameters. I will just explain that in a minute. And then we will integrate those outputs into the biodiversity results. So the term called integrative taxonomic approach, where we will be including the morphology, phylogeny, species delimitation approaches, right? And then there will be like ecology and biogeography, and then coloration and color patterns. So there are two examples which I would like to bring out from my study. One is the Saron species complex. What we basically did is there are two species. One is Saron marmoratus and then Saron neglectus, which is highly exploited for the marine ornamental trade. And these two have a wide geographic distribution in the Indo-Pacific, right? And then it has been a long taxonomic riddle, which has been not studied so far well. So I took this project as one of my, as a part of my postdoc project. And then I've started exploring this, and we did this based on the integrative taxonomic approaches. So these are some results coming out of it. Right, and we also incorporated more than morphology and molecular taxonomy. We also incorporated the coloration as well as biogeography in delimiting the species diversity in the Saron species complex. Another study is Lismata verdimani. This is widely distributed in the Gulf of Mexico and in the Western Atlantic Oceans. So the approach we undertook the same approach, where we differentiated separate clades, right, based on molecular phylogeny and morphological taxonomy. And we also did the haplotype network analysis as well as the population genetic, migration patterns, where how the species has been distributed and how the gene flow has got occurred. And moving on to the post-COVID-19, I would like to quote from Marie Curie. She said, like, nothing in life is to be feared, it's only to be understood. So I would say that it's again the time to understand more so that we will be fearless. I have like 4 points to shout out. Like one is we have to start to plan, since we don't know how the situation will be in the post-COVID. Maybe it will take another 1 or 2 months to get back to the normalcy. So what we can do is once we start move to the going on to the field, we can start collecting more samples, right? And then we can do the preservation for a long time, and then we can get support like from the collaborators, or as said by World Economic Forum, collaboration is the engine for global science. So if you look at my research, it will be more of like a, solo kind of a performance. So I haven't done majorly worked with several collaborators. And so if you are working in a collaboration, then we have to maintain the quality and ethics, right, to get the identity right, and then to make authentication of what we are doing. And then keeping timelines in the mind, priority should be based on the research, whether it could be taxonomy or it could be genome-based analysis. So it takes so much of time to do the whole genome sequencing or the mitochondrial genome sequencing. And then it takes so much of money, as Biju sir was mentioning about the grant related issues. And then if it is a ecology, again, there is no other option. You have to go to field, right? And then if it is a behavior studies, it will take few weeks to months, whether it could be a laboratory or it could be a field-based studies. And not but not least, of course, we have to depend on the joint publication. And then regarding funds, I would like to express that we can go for network projects, right? Combining 2-3 institutes and including their own expertise. And then maybe we can expect for some both national as well as international grants. So with this, I would like to stop here. I think I am under the time. Vijay sir?
Thank you.
Yeah.
Thank you, Prakash.
Yes.
You remain within the time and very valid points. And some of the panelists, if they want to ask anything, or and one point which you mentioned, Sanjeev, is very important, particularly with regard to the networking.
Yeah.
And there are some institutions where the networking is very much possible. And also, one should be rather broad-minded in this kind of an exercise, rather than in taxonomy. The openness and transparency is something very important, and also keeping the voucher specimens for every collection. This is also very important, because for many of the cross-checking, the voucher specimens are not available in many of the publications, and this poses a lot of issues with regard to taxonomic validation. And that's an important point. Anything other panelists to add? And there are some questions as well from the participants. James has raised his hand, and James, you wanted to talk? James?
Rajiv is having. Dr. Rajiv?
Yeah, please.
No, so I think Prakash does very similar work to the ones that we do. And one question that I have for Prakash is, he's been to all these universities and museums in the US. So how do you think these museums are coping up with COVID? So is there still work going on in these museums?
Yeah, exactly. They are still working on, and I would say even recently there is an issue from Australia. I think they are also looking for more funds regarding the museum, because they are unable to cope up with the financial crisis, especially for the taxonomic research. And then they are also unable to maintain the voucher specimens in the museum. So if I could say if developed countries are suffering like that, then it's better like a developing country like us have to, take precautionary step maybe in order to protect all those voucher specimens in India.
Because I think also for Dr. Biju, I think there is a lot of news that several museums are laying off people, collection managers, curators, etc. So I think
yeah, it has already happened in some of the museums, especially those on contract. They have been asked to vacate also in some of the places, and these things are there definitely.
So I think these are some of the challenges that taxonomists will face in the next few months, few years.
Yeah.
And James, are you asking some questions? James? Okay, then now.
Yes, sir. No, sir.
Okay.
Go ahead. Yeah.
Hello, sir.
Hello.
Am I audible?
Yeah.
Prakashana, hope you remember me. It's Ashik from CS.
Yeah.
My question is, how many markers are required to determine a species as cryptic species?
That's a very good question. According to some of the softwares, just mitochondrial DNA is more than enough. But if you want to validate those approaches, we have to go at least take 1 or 2 nuclear markers, and then we can go for a combined analysis, and it will definitely reveal the delimitations. And then we should not stop only with the markers. That's why I said we have to go with the integrative taxonomic approaches so that we can include other parameters like ecology, coloration, right? And then behavior, and all those parameters can be considered in order to reveal a delineate species. And if you go for just a marker, then mitochondrial DNA is very good.
Okay, one more question. One more. Yeah. Will permit one more question. Hello. Hello. Yeah. You tell your name and then ask the question, please. Hello. Yeah, you tell your name and ask the question, please. Okay, we'll go to the next presentation. Thank you, Prakash, for the nice presentation. We'll come back to you in the next round of discussion. And now Reshma Pitale is going to present. Reshma, are you around?
Yes, sir.
Yeah, and she is working with the Bombay Naturalist Society, and basically working on intertidal ecology and invertebrate taxonomy, and doing PhD in the taxonomy of polyclad flatworms or platyhelminthes, where none of the scientists in India is working. Reshma.
Yeah, thank you, sir, for this opportunity. I shall start with my presentation. Is it visible, sir?
Yeah.
Okay.
Make it full screen. Make it full screen.
Yeah. Okay. I'll be presenting a bit of my work about the polyclad flatworms and the challenges in the taxonomy of these worms, these beautiful worms, and how we can go ahead with these kind of invertebrate research in a post-COVID era. Just a basic about polyclad. These are platyhelminth worm. they have a many branched intestine, so the name is polyclad. They are leaf-like, thin, and maybe of a size of a few millimeter to a few centimeter long. The interesting thing is they have a very vibrant color patterns and beautiful worms. The relevance of this worm is these are some of the basal metazoans. So they have a unique importance in understanding the metazoan evolution, platyhelminth evolution itself. And they also owns the importance in the toxicology and the pharmaceutical branch, because they possess the tetrodotoxin, the TTX, the toxin very much similar to which is owned by the pufferfish. Ecology inhabit the different habitats like fish to coral reef to mangroves and the seagrass. They mostly predate on the compound ascidian sponges, corals, and some of the bryozoans or bivalves. Interestingly, as I have mentioned, they have a aposematic coloration, very bright color patterns, and some examples are available which also mimic the nudibranchs. I have not yet come across with any of such example yet. And also the tetrodotoxin, the point which I have already mentioned. About the diversity of polyclad, which was my first objective of the study. If you can see, there are only 7 families we know from India. And the first study which happened in India is about 1902, a British fellow Lakshadweep Islands. And then later there was no study about the polyclad diversity of India. In 2011, we could publish some species from the Lakshadweep, and then there are line of publications from Lakshadweep and Andaman and some Maharashtra. Right now, there are 8 species in the overall world, and we have only 64 reported species from India. So I think we can understand the huge gap we have about the polyclad diversity from our country. Regarding the study of my collection, I have got some interesting reports or the records of the species, I would say. Some are originally described from the America, from the Caribbean waters, from Brazil. Some are from Japan, some are from the Australian regions, and some are from Mediterranean. So we can see the interesting zoo geography I'm learning with. These are different species which we have found from the west coast of India, or from Maharashtra, I would say very precisely. Some interesting case studies which I would like to present apart from the other work which I could publish about the polyclads. The one interesting study is about this species, the Thysanozoon brochi. This is a cosmopolitan species. We can see its wide occurrence from the Atlantic. It was originally described from the Naples, Italy, in the Mediterranean region. And then there are multiple reports from the Atlantic waters till the Argentina, from the Pacific as well. And then some are from Japan, some are from New Zealand, and also some are from Sri Lanka and the Philippines. But all these are synonymized taxa, not the original reports of the Thysanozoon brochi. Like we can see the species was described in 1818, and after that there are multiple reports till the 2014. Under the name Thysanozoon brochi, we fell all that older literature and we could list out all that color maps, all that color patterns which are come under that species. And there were 7 original descriptions, as in named as a Thysanozoon brochi, published from 1818 to 2014. And we also could synonymize one taxa, one species, Thysanozoon lagidium, which was known from Brazilian water as a confirmed synonym for this species. But yet there are many theories. The interesting thing is that there are many theories of this species, because if we can see its distribution across the globe, across the marine regions, is because it's mostly in the tropical and the subtropical region. So how this species must have traveled? Is it a ballast which is causing, or it is a Lassepsian migration, or it is a species or a species complex? So there are multiple theories yet not able to confirm what it is exactly. The reason behind this distribution pattern, there are some researchers who is working from who is comparing the Mediterranean and the Atlantic Mediterranean, the Caribbean population of the species, but research is not yet published. So this is the one interesting thing about the polyclad research. And the another case which I have recently published is about the Stylostomum. The genus is got firstly reported from the tropics, while doing this species from India. There are taxonomic ambiguities about the assembly of the Stylostomum genus. There are a lot of inadequate information, confusion in the older description. So we could take this all these ambiguities in a same we could be what are those actual ambiguities in the anatomical features which are creating the confusion for the species identification or the generic determination as well, because there were different definitions of the genus as well. So presently, what I'm concentrating in my study is about identifying the homology between the anatomical features, because although the reproductive anatomy is important in identification of these worms, but there are still some of the homologous features which are, restricting the identification. So what are those other characters which we can still use to differentiate the species or the genera? But the main restriction is obviously the old samples or the older descriptions which are not inadequate to compare the species or the assemblage we are getting right now. And the next thing is obviously understanding the zoo geography and also the application of molecular analysis. As we all know, nowadays the molecular analysis is not just the differentiating the species, but there are many aspects which we can examine through the molecular analysis. But again, I would like to emphasize on these part, because I want the classical taxonomy and the correct combination of the molecular analysis to come together, so that we can also work on the classical anatomy or the aspects of anatomy of these worms. If we only concentrate on the molecular analysis, then we might we will never able to understand what are those anatomical features differentiating these species, if at all exist within the species or species complex. So these are the two aspects which I was concentrating right now. We haven't yet started with the molecular analysis. We are about to work on it. And the last important aspect is about the current scenario. So as I have portrayed about the polyclad, there are many hindrances like the less availability of polyclad work, not just in India, but in the Indian Ocean. There are huge gaps in the studies. The descriptions are scanty or not describing the characters which are important in the taxonomy, the species. So how we can address these issues? I think the questions which I have quoted about or the mention about polyclad is a case for many other marine invertebrate taxa. We haven't yet examined the significant amount of diversity or the marine invertebrate diversity we have in our coast. So if we can have the integrated approach where something like multi-taxa initiative we can come across, I think we have many researchers working in different parts of the country. If we can take them together, pool the efforts together, and work on the multiple taxa or the selected list of taxa for the taxonomic approach. This will include the classical taxonomy as well as the molecular approach. And the basic things which I would again I will like to highlight is the habitat and the species conservation goes hand in hand. If we have more data about the habitat and the species, we can also use habitat conservation as well. So I think the huge efforts on collection which all of us require, and when there are less funding opportunity or the scenario like this where we may not travel across the country because of this kind of scenarios, then I think we can pull together all these efforts where multi people will be involved, multi taxa will be involved. So there will be more data and more information about these neglected group or neglected marine invertebrates.
Thank you.
And, again, we also can achieve something called as conservation of habitat.
Thank you. Thank you, Reshma. Thank you. Thank you for the presentation. And one important aspect here, which Reshma raised, is definitely very much valid, and that is regarding the multi-taxa survey. If I am not working in a particular group, I go to the field and collect a mini organism. And definitely,
Yes,
with the
yes.
collaborative groups are there, you can share it with them, and that is the way in which we can, when there is
Absolutely.
deficiency in funding, definitely we can
Yes.
increase the output of research from India. Any
Absolutely.
panelists? Yeah.
Exactly.
And Roshni has raised the hand. Roshni, if you want to ask something. Roshni? Hello? You have to unmute and talk. Jofin? Jofin? Okay, any other panelists want to ask something with Deepani?
I just wanted to, sorry, can you hear me?
Yeah.
This is Dipani. I just wanted to know if they were an indicator species of ecosystem health in any way. Do we know any such
No, madam,
details?
no, not yet. But they do feed on, they voraciously feed on corals or some bivalve species. I think that much of information is known about their ecological points.
Okay.
Okay, sir. Dr. Sarangi?
Yeah, good evening to all panelists and participants. I do not exactly work on this topic, but I have common queries, basic queries. What about the pharmaceutical importance of this species, Platyhelminthes polyclad? So, is it antibacterial, antiviral, or what kind of pharmaceutical importance? And other additional queries that life cycle, what is the lifespan of this Platyhelminthes polyclad?
Sir, for life cycle, information is not available. There are, I think, 2 or 3 studies from our country which from where they have brought the animals to the laboratory and kept live. But the maintenance is really crucial because they are really sensitive species to the environmental or the water parameters. But the second thing is in terms of pharmaceuticals, they have a TTX which I have mentioned. So they are using it and extracting, not really extracting on the what I can say commercial scale, but they are experimenting it from different for different purposes, like maybe for some examples for cancer and some other aspects, but nothing on the commercial scale about the research which is happening about.
Okay, I think a lot of research required on this particular taxon, particular species, particular
Yes,
genus.
sir.
Okay,
Yes, sir,
thank you.
definitely.
Thank you.
Dr. Shibu, want to talk? Shibu?
Dr. Vijay Kumar, I just wanted to
Sure. Yeah.
ask something.
Yeah.
Can I? This is Dr. Ingole.
Yeah.
Reshma, compliments to you for a good, very interesting and very good work.
Thank you, sir.
Best wishes for your PhD, because it is not easy to work on such organisms. First and most important, locating them, and then identification when you don't have much of expertise available in the country. So compliment to you. And I just wanted to know, certainly there are other people who are probably watching this webinar. They may have some samples in their collections, or even future they might be. Will it be possible for you to identify them or collaborate with them up to the extent whatever is possible, so that we can, give the message, because this is the area, as Dr. Vijay Kumar in the opening only, no one is working in India, and therefore I thought it could be, good to start with. At least we have someone, and let us make use of it. Why I'm telling you, back almost 4 decades, in 78, 79, I did my MSc dissertation on the, the turbellarians, and I know after that hardly there is any work on the ecology, culture, and all other aspects. And
Yes.
this is a fantastic group of organisms. So
Certainly.
that's what I just wanted to.
Sure, sir. Sure. I will try my best. If somebody can bring samples and we can identify them to the possible taxonomic level. Definitely.
Okay, thank you. Dr. Shibu, you are asking something? No? Okay. Okay, thank you, Reshma, for the nice presentation.
Thank you.
And we'll move on to Dr. Jabir. Jabir is the project scientist with the National Center for Polar and Ocean Research in Goa. And he is actually a marine microbial ecologist, as well as he work on marine microbes. And currently his research is focusing on microbial diversity in the extreme environments. Again, not many work in this field. Dr. Jabir.
The screen is visible.
Yeah.
I am Dr. Jabir Tajuddin, working as a project scientist at National Center for Polar and Ocean Research, Goa. I am working on the microbial diversity of the extreme environment. Earlier, I worked in the Arabian Sea. Now, I am working on the Arctic environment. Today, I am going to discuss about the microbial diversity and the recent research and the process in the post-COVID era. All of you know that microorganisms are invisible to human. They are very small and abundant in the ocean. About 10-30 cells are present. About 10-30 cells of bacteria are present in the ocean. They are in one sample. There are 10-9 eukaryotic cells present. These organisms are a long history and being our planet to the lack environment. Recent studies on the bacteria, recent studies on the microbiome have in the new paradigm because these are basic unit of life. They are the producers. They are doing many biogeographic study of these organisms. Study of these diversity functional role are very prevalent in this. So, how we can see these microbes? Earlier we worked with the culture dependent and culture independent analysis. Culture dependent analysis means to platform method. Next level, nowadays we are doing the metadynamics. So, microbial first, mainly on the bacteria growing on the plates. But only 0.1% of marine organisms are culturable. The rest of them are non-culturable. So, in the genome in 2004, from the Hydro lab, they use the shotgun from that DNA sample. They are identifying the accurate microbial identification. The first
Jabir, your voice is not clear. Jabir, your voice is not clear. Yeah, there's a lot of break in between.
Now it's clear, sir. It's okay.
Yes, now it's clear.
In 2004.
Yeah, now it's kicking.
Now it's okay, sir.
Yeah.
Okay. These microbes our first metagenomic study evolved in 2004. The highest, about 1.2 million DNA. Their first study, they identified nearly 200 new species of organisms in genome. Then the exploration of identification of these microbes. They are in metagenomics, genomics now various studies are going in from 2000 to 2019. Very large data set of microbes are available in the world, especially Tara Ocean data set, international census of marine microbes, global ocean sampling data, world ocean sampling data. Many databases are available. In 2015, from this Tara Ocean data set, we first created this ocean data set and highly evolved, especially bacteria, archaea, and virus through sequencing. Complete comprehensive biodiversity reports in Science 2015, a special issue on that. But in our region, especially in the Arabian Sea, only limited data is available. In the ocean, in total world ocean, very good databases is available. Through this database, we can do a lot of things. We don't know. We not comprehensively. We have comprehensively analyzed these data, but we have to identify the role of these microbes. We have to identify their biology, their potential. But with these data set, we can go further. So, in this data set, Agatal 2016, they conducted a new microbial lineage, especially bacteria candidate radiation. These group of bacteria.
Jabir, again, there is an issue. We are not able to hear you clearly at all.
Okay.
Please check your audio once, yeah.
Hello, now it's okay, sir? Okay. These candidate phyla radiation, you can see these candidate phyla radiation, these are unculturable organisms, but we only know the sequences. Currently, through metagenomics data, we only know the sequences. As like bacteria, earlier we know only green archaeota and new archaeota, but currently through metagenomics data, about 26 archaeal phyla, but these archaeal phyla are also uncultured. But nowadays we know the sequence. These sequences we cannot go to physiology and other function of these organisms. We don't know, but we don't to culture these organisms, and also from this database we don't know more about the microbes. So with this database we can go further and we can study the biogeography and the functional process and the functional potential of these microbes and their post-climate change and post-COVID era. What are the climate change implications and the environmental significance of these organisms? Yes, sir. Thank you, sir.
Thank you. Thank you, Jabir. So there were some issues with the audio in between.
Okay, sir.
Now, Ravi, Teja, you want to ask something? Ravi? Ajita? Ajita? Hello. Now, those who are raising the hand are not asking questions. Biplab Maji. Mr. Maji.
Hello, am I audible?
Yeah.
Sir, my question is, what are the economic impacts of marine litter?
Hello.
Hello.
Please pardon. Yeah.
Am I audible?
Yeah.
What are the economic impacts of marine litter?
Economic impact.
Yes.
Impact, marine litter, recent studies on the antibiotic resistance is marine litter form new antibiotic resistant side. They are from the marine litter, we are taking these bacteria doing more antibiotic resistance. That side, because it's a COVID era, next level, antibiotic resistance is a major issues. So next level, through antibiotic side also we can study in this marine litter. Yeah.
And my question to Jabir is that, when you study extremophils, in addition to bacteria, what are the common organisms you come across, and
extremophiles, now we are focusing on the archaea. Archaea, we don't know more about the archaeal sequences and archaeal phylogeny.
Somebody in India is doing that?
No, sir. Especially recently, nearly eukaryotic phyla, Asgard archaea is the new phyla recently discovered, but we are currently working on that. But somebody in India, nobody is doing. India microbial oceanography also still in rudimentary stage. We need to study more. We need to develop more.
You all have the facilities for metabarcoding in your lab, or?
Yeah, we have, sir.
Do you have any collaboration with other institutions?
Sir, I have one.
Sir, please pardon?
Any other collaboration going on with other institutions?
Currently, we don't have any collaboration with other institutes. With the Norwegian, with the NPA only, we have one collaboration. With the we need collaboration with some other agencies do the next level metagenome sequencing.
Yeah, but why I asked this question is that, I was told from the National Biodiversity Authority that there are a large number of requests for permission, particularly for the microbes. The transfer of materials with regard to the sequencing as well as other genetic studies.
Yeah.
Okay, thank you, Dr. Jabir, for the presentation. And now I ask Dr. Dinesh to make the presentation. Dinesh? Dr. Dinesh?
Yeah, I'm going to share it now. I'm audible?
Yeah. And Dr. Dinesh worked with the Department of Microbiology, School of Life Sciences at Pondicherry University, and is a coral reef ecologist and a former Fulbright postdoctoral fellow, working with and his research focused primarily on coral reefs and its association with climate change. Yeah.
So my slide is visible right now?
Yes.
Okay. So thanks for the well-created platform to share our research to everyone. Thank you, sir, for that. And today I'm going to present the current threat, what our coral reefs are facing, and what are So this is my overall talk roadmap. So here I'm going to share 3 of my experiments I did in order to conserve it. So when I was doing PhD, we surveyed so many islands and so many coral reefs. We found sponge overgrowth and coral disease was a major threat. So that's the first issue we found it. Then after my PhD degree, then I want to work on coral bleaching, like how corals are adapting to the increasing temperature. So what are the experiments I have done? I'm going to share it with the second one. The third one, what is the role of symbionts in coral transplantation? Why do we really need to care about symbionts in coral transplantation? So these are the roadmap I'm going to talk about. So before I enter into the story, I just want to introduce some of the important characters involving in this story. Okay, so we all know that corals are interconnected coral polyps that construct this mega structure, intricate relationship between corals and the symbionts, as well as millions and millions of bacteria and viruses. So these viruses and bacteria, symbionts playing a major role in constructing this mega reef. So as I mentioned earlier, the hollow, this is one kind of polyp. You can see a lot of viruses, bacteria, symbionts, and importantly, dinoflagella sitting inside the coral. That is so interesting. So as I mentioned, this is one picture. It's taken from the fluorescent microscope. It's clearly visible like how symbionts is accumulated in the coral. So why symbiont is so important is that fulfills the coral's energy need. If symbiont is not there, the corals cannot survive. That's the basic stuff. So as I mentioned earlier, the corals are hosting symbionts. That too, like not only single symbiont type, it has many symbiont types like C, D, B, A, and different type of symbionts with different composition in order to adjust to the different environmental conditions. Okay, so threats to the coral reef, as I mentioned earlier, sponge overgrowth is a serious threat. In Park Bay area, it's killed almost 80%, almost 60% of the corals. And coral bleaching, as we all know that it's a serious issue. Okay, so this is the experiment I did. Like initially when I looked at it, like it's spreading, then I wanted to look at how what all the species got affected. Then I started to tag each and every colonies I encountered. Then I found it's really fast than what we anticipated. Like it's more than 3 centimeters grows per month. Just imagine if it's a small recruits to kill the top of the coral recruits. So these are some of the images I took. Like coral sponge is really threat. And this other experiment I found, like how speed it is. Like first colony within 3 months of time, within 6 months of time, the entire big size colonies died. It took around, the entire colony to grow, it's around maybe 10 to 15 years to take. Okay, this is one of the survey we did in August 2009. The sponge occurrence was very less. Then at the end, it's killed, almost 80% of the coral in our study site. Okay, then we thought, okay, what we can do? Okay, the sponge is spreading really faster. It's killing corals like anything. What we can do? Then we came to know the recent paper. They mentioned that sponge is also having great symbiont association with one type of algae that is getting nutrients from the sunlight. Then that progress will really faster and kill the corals. Then we thought of coming with the experiment that, okay, what can we share the sunlight so that the sponge cannot grow really faster? Then we shared it with the light, and then this is the experiment we set up. This is a control site. Here you can see after 6 months of time, the entire coral colony died, but where we tie the cover, the coral we saved. Then so that paper got published in past one. Then work in progress. Like you can clearly see that's a clear cut difference. The white color. Now we are wondering what kind of microbial competition is happening in there, because corals also hosting coral symbionts, bacterial community. Sponge is also hosting coral bacterial community. So initial fight should be between the bacteria. So we are collected samples between both the sides, and we are looking at what kind of mechanism is happening over there. So now I'm moving to the second objective, second experiment, like bleaching. As we all know that bleaching is a serious issue. It's spreading coral like anything. It's killing coral like anything. One of the recent study predicted within 2050 we're going to lose most of the corals all over the world. So as I mentioned earlier, like normal coral will always host so many symbionts in it, so many symbiont types. And when it's bleached, actually the bleaching is a phenomenon when the temperature is going high, the symbionts not able to sitting inside the coral, it's going out. So ultimately the coral lost its color and it died. But fortunately, some of the coral is able to survive well by changing its symbionts. So my experiment is like how it is doing, what are the composition required for the coral to adapt to the increasing temperature. So bleaching hypothesis suggests us that like sifting and suffling is a two process corals are doing in order to survive in the post-bleaching condition. So in India, before we started this work, like we did not generate that many information about the symbiont type, what type of symbionts corals hosted, and how it is adapting, how it is changing. Then we Then we thought of, we went for the survey, then as we expected, we found a clear difference between the species and within the species. You can clearly see in the picture, there is a clear difference, even both the species are sitting close together. Okay, so these are the other pictures we have taken when we go for survey. Like there is a clear pattern in the differential pattern in the beach. So this is the site we selected for the parkway. We got a permission to work on it. So this is the experimental setup we did. So bleaching condition, we selected 4 species: Acropora, Favites, Favia, and Symphylia, because these species are dominant in the particular location. Then we have collected, DNA, and we looked on it. And then after bleaching, what are the species recovered from the tagged colony? Then we collected the resampling again the same colony. Then we found that the corals in our Indian coast is mostly hosting Symbiont Glade D. Before bleaching, Acropora hosted only C. But after bleaching, all the coral species changed into Symbiont D. Okay, so why Symbiont is matter? Because Symbiont Glade D is one of the species that helps corals to adapt really faster, but Symbiont C helps the corals to construct the reef really fast. So what we have right now, we have Symbiont type Glade D. So our reef is adapting, but it's not growing really fast. That's a real concern that we need to look at in so detail. So in that work, like what we really need to look at is, like in my work, it shows like what is the abundance of the particular type. But what we really need to look at is like what is the composition of it, because even cryptic species really matter. Before bleaching, if it's cryptic species smaller, then it is changing to adaptive species. So those are the things it needs to be looked into. So moving on to the third experiment I did back in USA is, okay, how the symbionts is playing a major role when we do transplant the corals into the different environmental conditions. So right now, the ecologists, the coral transplantation, it's happening all over the world. The major problem is like I mean, I agree that transplantation is really hard job. It's not easy. But what we're doing is like we transplant the corals in the shallow water area, not more than 10-meter area. So we thought of transplanting the corals the deeper area where people easily cannot go, right? So that we can use that preserved reef to repopulate the bleached corals in the particular location. That was the rationale behind our study. So as I mentioned earlier, the bleaching is a serious issue. If you really work on it, I mean, invest a lot of time, work on it, the transplantation, but what happened, one simple bleaching, it's lost out everything. So yeah, we did. So we decided to work on the 2 species, Orbicella and other species called Orbicella species. Other species, the Orbicella, different species. So why? Because the Orbicella is highly endangered right now. Few colonies, maybe we can count like 800 to 1,000 colonies sitting in the entire Florida coast. So we decided it. So we worked with the NASA NEMO team. Actually, they worked for analog missions. So they helped us to construct the deep sea nurseries where we cannot go. So they constructed the nurseries in the deep sea, like 30-meter sea. So this is approach of our study. We selected genets. So we selected different genets because each genet plays a major role, we thought. Then we cut it into the small fragments. Then we tied into the small ID card stuff, and we constructed a tree inside the reef. So this is the site we collected the reef. Coral colonies. Then each and every branch is hosting different genotypes. So the question is this: like how our what is the role of symbionts in adapting to the different temperature? This is the experiment setup we did in Conch Reef. We did 10 meter, 20 meter, and 30 meter. So 30 meter only they did for us in order to collect samples. Oh, sorry. So after that, 1 year we kept the sample inside the water. Then we collected the particular species. Then we categorized the genets into what are the genets is growing faster, what are the genets are not performing well, what genets are not performing well, and what genets are affected by overgrowing competitive organisms and all. So yeah, at last we found like overall in genet 13 really perform better than other genets. Based on that, they decided, okay, they started to preserve that particular genets. So right now, as I mentioned earlier, there are so many symbiont types. If C is hosted, that helps to construct the reef. If D is there, that helps to adapt the reef. So now we are analyzing all the samples. To look at when we take the corals back into the deep reef and back into the bring back to the shallow, how the corals are helping to adapt it. Yeah, this is what my work so far. And regarding post COVID era, so as young researchers, we need to have some projects in our hand to work on to carry out this research forward and achieve the results. I mean, I personally feel like in India, in the medical field, like we do have excellent facilities in terms of sequencing, histopathology, and all. But if we go into the marine biology section, like I feel we are a little bit far behind when we compare with the molecular work when we compare with medical science people. So I need that kind of collaboration. If it starts, that's going to be really take our research in a little bit more forward. And one more important point that Madam initially mentioned, the webinar. Previously, it was not happen like this. It's a great opportunity to attend meeting without any fear and talk with many people. So I hope this kind of thing will keep continuing and will work really forward. Thank you, sir.
Thank you, Dinesh, for the excellent presentation.
Hope I finished my.
Yeah, it's okay. And one question is asked by Riti Sharma. Riti, you wanted to ask questions? Riti?
So once done, I have a couple of questions.
Yeah, Deepak. Okay.
Can I go?
Please.
Dinesh, I am seeing you from the time you joined as a researcher, and the way you've grown. Compliments to the work that you've done. I have just three simple questions. Okay, do we have a resilient species for India? Number one. I'll just finish my questions, then you can probably answer. The second one is that see, we are also talking about diseases happening in reefs. Okay, is there any specific reef to soft corals? Because that is one place that is really neglected.
Oh, yeah.
And my third question is that the Orbicella genus that you showed me in the US is almost similar to the Porites in terms of morphology. So is there a possibility to replicate experiment like that in India?
Yeah, man, three questions are super questions. I will answer it one by one. So the first question you asked is, okay, what is the most resilient species?
Resilient, yeah.
Okay, so based on my observation and my colleagues' observation, we found the favorites is one of the highly resistant species against the bleaching. So far, like in my work previously, I found even all the species bleach, even Symphilia bleach. Symphilia, that's one of the massive species bleach. The favorite is resilient. So yeah, that's a resilient species, I would say. Did I answer the question?
Yes, you did. Yes, and second is about the soft coral, and the third is about the Orbicella. No.
Okay, so the soft coral, like I agree completely. I mean, 100% I agree with you. The soft coral is one of the important taxa. We need to really document it first. Then we need to work on it. I mean, being interested to work on symbionts, I'm really curious to know what symbiont type is hosting there. Right? SDMRI people are doing a documentation part in the soft coral, and NAO people are also doing some documentation work in West Coast, I guess. Yeah, that's what about. And the third question is
Orbicella.
Yeah, Orbicella. Orbicella, I would say it's a little bit. I mean, in terms of polyp structure, it's a little bit different. So Porites polyp is tiny, but it's intruded. I mean, it's intruded into the skeleton. So in terms of resilient, it is stronger. But in the case of Orbicella, it is different. So the Orbicella is not construct like a mega massive structure like a massive coral. It's always encrust over some other massive stuff and growing it. So if you want to look at if you want to replicate the same thing, I would suggest I would select the what's called that Turbinaria species. Yes, Turbinaria is more or less similar. The polyp size is almost similar. Yeah, that we could definitely choose it for.
Thank you, Dinesh. And one more question, Ritika Sharma, you're asking questions, Ritika Sharma. Ritika? My question to Dr. Dinesh is that you have an experience with the US systems, and how the linkages are continuing after you are coming back to India.
Yeah, I mean, I keep in touch. So far, I keep in touch. So we are planning to start a new project, like how how our Indian reefs are doing, and when we compare with that reef, like what is Symbiont type and what they are doing, when it's recovering, how it is changing. Those are our plan right now. But I mean, to be very honest, I'm really looking for a place to sit down and to initiate all this thing. But right now, I'm seriously working on the paper I worked on and writing down.
Okay, thank you, Dr. Dinesh. Thank you. And next is Dr. Sanjay. Hello, Dr. Sarangi, Dr. R.K. Sarangi, and he is actually there amongst us, the taxonomists, mainly to add a flavor, and how the remote sensing technologies and the work of ISRO also can help the youngsters in bringing out good work. And you can explain.
Yeah,
Dr. Sarangi.
Yes, sir. Okay, so I am the fifth participant. Means I have to tell about the ISRO program. Is it?
Yeah.
Hello. Yeah.
Yeah.
Is it audible? Yeah. Yes. Actually, because this International Ocean Day, this forum I had came to know just 2-3 days back. Then immediately I thought I should join, and because a lot of participants panelists I know, even Dr. Babu Ningole I know from long back. Then even Dr. Samuel, and then so many important topics also covered will be covered. Because as I understand, it is our cup of tea, because my primary subject. Then I entered to remote sensing satellite applications. So that way I thought it will be very much useful for the participants and for me to have this exposure. So then immediately I thought I should register and continue. So as we understand, this remote sensing has a lot of potential nowadays to utilize these resources, marine resources, and this climate change, then this ocean processes and ocean features. There are several coastal features, and like India is influenced by this variety of cyclones, upwelling, then river fluxes, and this convective eddies. So many other features are there in larger scale, broader scale. So, but we need to link to in smaller scale in regional seas how these ocean features and oceanic phenomenon are impacting to our ecosystems in small, larger, wide, broad scale to even to kilometer to meter scale, and even to very narrow scale. So how it is impacting in different time scales, that satellite data we are trying to analyze and synthesize. So that way I found these upcoming researchers and students should propose different ideas, different projects. So to link to different even their biodiversity. In terms of, there are some good presentation there in this platyhelminthes, and even corals. There are a lot of study going on, and proposed new activity. I liked very much this Dr. Dinesh, how he presented. So his work on this related to NASA, this NEMO mission of Martian analogs, that lunar analogs, that planetary analogs data, they are trying to probably link to this coral reef and extreme environments, extreme waters and extreme conditions. So that way, so exposure is required for students to have this remote sensing satellite, because we have this satellites from this optical, then microwave, then radar, then hyperspectral, then we have altimeter, scatterometer, micro radiometer, thermal infrared radiometer. There are several technology we have. So that way I feel all these different type of satellite sensors, how this can be applicable to this marine scientists, and overall this marine biology can be strengthened with use of all these variety of satellites. So that way I feel it is required to have these young researchers to come forward and try to have their their research activity in a larger scale. So will be useful to this marine community, and this satellite technology and remote sensing community will be very much grateful to marine biologists. So that is my means request young researchers to have these new proposals. Advanced remote sensing satellite data can be utilized, like this hyperspectral, and even radar also people are trying to utilize in this like mud banks, mud floods, variety of means environments can be linked. So that way my suggestion and request to our young researchers teams to link to all these spectral channels available using different satellite, different sensors, Indian satellites as well as global satellites, so that our all these marine environment like benthic, pelagic, then I think all these even bottom environments, coral reefs, mangroves, all coastal environments that can be utilized in larger scale. So that's way I feel our data, satellite data, in even in COVID scenario, COVID post-COVID scenario. So this environment, how pollution is taking place. So its response to these microbes, to including to microbes, then ecology, and then fishery also is important aspect. How this our fishing resources, whether it is disturbed or not, and how it is important to have this information for using satellite data, like all this PFZ activities means already inquest is taken off since decade, more than decade. So how this can be linked in the broader scale using different satellite applications technology. I hope this will be very useful for the students and participants to have this utilizing satellite technology, how they can use this their subject knowledge, and including this variety of marine biology aspects, like even cytology. I saw some work on this genomes of prawn, some shrimp genomes also. Somebody, first participant, I think Dr. Sanjeev, he has shown. So similarly, like remote sensing point of view, different satellite technology, all these channels I told, the spectral, like optical, thermal infrared, near infrared, and microwave, radar, then hyperspectral, altimeter, microwave radiometer, scatterometer, everything can be synergistically utilized, so that this fishery forecasting system also can be means to be very much advanced, so that currently only SST, chlorophyll, and other wind, other thing parameters is used, so that larger parameters like mixed layer depth, and then optical depth, and even these nutrients, all these nutrients, nitrate, phosphate, sulfate, and different means constituents, sediments, TSM, all these carbon primary productivity also what is happening. So variety of parameters needs to be correlated, interlinked, and this impact on this marine ecosystem, how it is happening in this on this various biogeochemical cycling, that is also another challenge. So that I request all these participants, those who are means attending, so they should be able to utilize our technology, and how it will be very fruitful means in larger scale and in long term. So that is my means observation and my comments. Thank you.
Thank you, Dr. Sarangi. Dr. Baba Ningole Saab, you have something to supplement?
Yes, very much. Dr. Sarangi, thank you very much for your
Yes, sir.
very good point you have raised, and I would like to request some of the youngsters. Basically, this is a good opportunity for all of us. Post-COVID, there are a lot of restrictions going to be there, as in the beginning Dr. Biju Kumar mentioned it, funding, field work, and also we will need a lot of collaborations, particularly networking, and for that purpose remote sensing technology will be very handy. We can collect a lot of data in a short time, and that can be used by a group of people. Particularly youngsters should definitely come out with the proposals where remote sensing can be useful for collecting the long-term data, and on the broad scales data and lot of parameters that as Dr. Sarangi has mentioned it. But maybe we'll have to sit and then we'll have to work out how best we can come to this one. Thank you very much, Dr. Sarangi, for that.
Yes, sir. Thank you. Thank you.
Thank you, Dr. Sarangi. We will move on to the next
I
presentation.
have finished.
Yeah, sir, something more to say, Bhavan sir?
Go ahead. Go ahead. Go ahead with the.
Yeah. Next presenter is Dr. Ravichandra Mondreti, and he is working with Department of Ecology and Environmental Sciences, Pondicherry University. He is actually working on the ecology of seabirds in Lakshadweep and Bay of Bengal, and is deeply passionate about seabird conservation at the grassroots level. Dr. Ravichandra.
Good evening to all. Hope I'm audible to everyone.
Yes.
I'll just share my screen with you all people. Okay. Sorry. Okay. Good evening to everyone. And good evening to Dr. Biju Kumar for inviting in this in this event. I'm very glad that I'm presenting my work here. So actually, what I'm going to present is my my PhD work, which I have carried out during very recently. I've done the work on the seabirds. Coming to, I worked on the ecology and conservation of seabirds in Bay of Bengal and Lakshadweep. As such, it's a very broad study. Though it's a broad study, my research objectives are very confined to a uh, certain sections. So, which I first of all, coming to seabirds. In India, actually, we don't have much work done on seabirds. In fact, there are, except for few opportunistic records, so nothing much has been carried out until now. But as such, I would say seabirds. Coming to the ecology of seabirds, actually, seabirds are highly visible and it can be easily observed, from the colonies. Even there, and they because of this colonial behavior, since this is an intrinsic property of them, so intrinsic behavior. So and seabirds are very easy to monitor. For example, they exist with other species, and there are numerous colonies around the world. In fact, in Seychelles and all, where they have identified somewhere around more than 50 species. No, in fact, and also one most important thing why seabirds are very good in like good monitoring species is basically they're like they can be studied in a very in a very no, very good platforms. For example, you can do ship surveys for observing while counting the birds through sea surveys, and also through colony monitoring studies. And they can, in India, actually, they're highly understudied species. And coming to their role in marine food webs, they occupy an important role along with other marine predators. And they share like sharks and other cetaceans. And my study actually addressed certain gaps in knowledge on seabirds. So I focused my research on on certain aspects which I have found while going through the literature, and as well as while doing research on the all other studies which have been carried out previously. So what I found is basically in Bay of Bengal we have little data on seabird sightings. As such, even though there's sightings available, but they have been done on opportunistic basis. While they were when they were carrying out other studies, they have additionally observed seabirds and then recorded it. So other thing which I found is in fact there were no comparison between different, for example, different comparative studies between India and as well as other in other parts of the world. And this is my study is also a preliminary attempt in knowing the seabird distributions. Hello, am I audible? Hello, sir. Yeah. I'm sorry. Yeah, actually, I'll just continue forward. this is also a preliminary attempt in recording the seabird distribution using systematic survey techniques. So for this study, I collaborated with one called Dr. David Gremier from Center for Ecology and Evolution function in France. So I also collaborated with Dr. Peter Ayan from University of Cape Town. So he was actually Dr. David Gremier was co-supervisor of my PhD work. I carried my no my PhD study under Dr. Priya Devidar. So Dr. When we decided, we have focused in fact we designed a study of observing seabirds at sea, so using the passenger vessels. So also the main other in fact gaps which I try to address during my study is like to understand how the seabird communities are structured in Bay of Bengal. So at least though my study is not very exhaustive, though it's like exploratory, so I try to understand the patterns. So how in fact in what way they're different from other parts of the world. So this is like completely a novel attempt. So I also try to understand whether there are any interactions between pelagic longlines and seabirds in Bay of Bengal. So even though we couldn't make much direct observations, so what I try to assess is from the both like all the records available on this longline pelagic longline fisheries that which I've taken from IOTC, Indian Ocean Tuna Commission. So I also as a part of the study, I also wanted to document there is this seabird breeding colony in Lakshadweep that is in Piti, very close to the Kavaratti Island. So here actually this is the most well known colony, well known pelagic seabird colony in India. So and they have seabirds have been breeding over many decades over there. Like it's been happening over ages. So it's not decades actually. So many centuries. So and I tried to document seabird colony over there. So in fact, they were also like I also try to understand the attitudes of people towards seabird conservation, but I won't be discussing that much in my during present talk. So what first of all my preliminary objective of my study was I wanted to establish a baseline information on the distribution and abundance of seabirds in relation to environmental parameters. So, which for environmental parameters, I choose sea surface temperature and chlorophyll A data. So, which is a proxy of primary productivity. So I also chosen bathymetry as indicates, it predicts the seabird distribution to a very, in fact, good level. So the second objective of my study was to, in fact, analyze the spatial overlap between foraging seabirds and pelagic longline fisheries. For this, I used remotely sensed data as well as the data available on the pelagic. Different longline pelagic fishing data sets available with the IOTC. In fact, the third objective, which is like I also studied on I wanted to create a baseline information on the pelagic seabird colony in Lakshadweep. The main species which are breeding over there is sooty terns and brown noddies. So what I have done during my study is like I've documented the population size. So I've carried out many colony monitoring studies. So over a period, so I recorded what are the species and what are their number, what is the abundance at regular intervals. So, and also I have observed in fact. Certain events of egg harvesting. So people have been harvesting, people have been collecting these eggs over a long time. So that has been happening as of yet. I would say like I also tried to enumerate like to at least to create a baseline. So what is happening exactly during this collection of eggs in order to assess the threats to the seabirds. So regarding the at-sea survey which we have carried out, I choose a predetermined transect. So I basically did it by I used like both the passenger survey, research vessels as well as research vessels. Sorry. So while the passenger vessels, I choose the basic route from Chennai and Kolkata, the two fact services have been operating. So I choose that, and then I did the transact. So and I also there was like other collaborators who have utilized the research vessel, and they have done a more elongated transact in Bay of Bengal. These are the different platforms we use, like both research vessels as well as passenger vessels, and we observed seabirds using the platform from the pilot house in the ship. So these are basically my results. So I'll just show you these are the maps of the distribution of the seabirds. So I've just in fact projected the distributions on sea surface temperature, and then I wanted to see whether there are any patterns. So I generated the patterns. So these are the basic maps. So I also did the same thing for chlorophyll primary productivity. I observed both. I happened to also record citations during my study. So I also basically included as a part of my study. So we have also opportunistically recorded citations during our study. So even I also seen whether there is any relation between bathymetry, especially the depth and the seabird distributions. And finally, what we have done is like I've created maps, fishing effort maps, and then projected the seabird distribution on that to see whether there are any overlap, areas in Bay of Bengal. So this is the final part of my study which I've done. Like I have actually tried to document the whatever baseline information I have generated on the on the seabird colony in Lakshadweep. So these are my study area. So I've chosen PT Island. So basically this is the only well documented colony, well known colony of pelagic seabirds. I monitored seabirds in the I visited the island. So I using a scope we monitor the seabirds. So this is on the right is actually the publications, recent publications, which in fact one of which is very recent. So I have recently only published that about seabirds and citation occurrence. Coming to the how we can carry out during this post-COVID era and all. So what I found is uh, if you're doing a long-term monitoring study or any monitoring study as such, and if you're working on some remote areas, so I think it will be good if we employ some remote survey techniques like employing UAV, UAV technology. So I think that would be for even drone technologies. That will be really helpful. In fact, for people who wanted to do long-term ecological monitoring studies. So and also it's also good if you involve local communities as a part of research data collection. So it's good like we train them, and then when they get trained, so they will be of very helpful to the researchers. Which otherwise during this post-COVID, it will be very difficult to access the sites. And also encouraging more citizen science initiatives, though people can collect whatever possible data available in their localities itself, so without having to without researchers having to go all the way, and then and also encouraging more collaborations between researchers as well as non-researchers. So I would like to end my presentation here, and thanks for giving me the opportunity. So thank you very much.
Thank you, Ravichandra. One question by Mr. Vishwanathan. You want to ask question? Vishwanathan.
Hello.
Yeah.
Hello.
Yeah.
Yes.
Can you able to hear me, sir?
I can hear you, sir. Yeah, please.
Yes, sir. Thank you for your presentation, Dr. Ravichandran.
Thank you. Thank you very much.
Very nice work. But the one thing I want to ask you that
Yeah.
why you compare the bathymetry data for your study? What is the relationship between your study, means how the bathymetry data is related to the seabed population or the study? I want to know.
Yeah, bathymetry is actually a good indicator of seabed distribution. In that way, in the sense, since in tropical areas mostly this you will find across the surface diving seabirds. So actually bathymetry is a good indicator of fish shoals. So, and people have been using this bathymetry data to locate the fish shoals, especially people who are in the like in the sense in the field of pelagic long line on fishermen, usually employ this technique. So as such, like though remote sensing data may not be like in the sense an exact it may not generate exact data, but still it's a fair good indicator of seabird distribution. So seabirds usually follow these fish shoals, especially they
Okay,
keep on following that. So yeah,
okay.
hope I have
Basically,
yeah.
I'm working in ecological sensitive areas and biology and somewhat in taxonomy.
Okay, sir.
But I have somewhat how to say, I have less knowledge in bathymetry, and
Okay, sir.
I know that bathymetry is mostly dealing with the water current and movement. My knowledge.
Yeah, okay.
That's what.
You're very right. Actually, the thing is that in tropical, environments, especially in tropical marine environments, so what happens here is actually seabirds have to depend on other predators like tuna, and basically other cetaceans like bottlenose dolphins and spinner dolphins. So they all form good association. So and then bathymetry as such, like as you said, since as good indicator of currents and all. So these fish usually this follow the currents. So this fish movement basically, in fact, they keep on following according to the currents. Their movements, usually depend on that.
Okay,
So and these.
So
thank
can
you.
we take?
Dr. Sarangi?
Yeah, yes, sir.
Yes.
Actually, very nice work by Dr. Ravichandra. So it is like we also are interested to have such kind of collaboration with him in future. And this I have query regarding why you have not tried to use this wind data, wind vector, wind direction, and that is very crucial parameter. You should be able to get this their pattern of their migration pattern or any other movement that I think would have been suitable parameter along with chlorophyll SST. Okay, like other param means fishery and other marine means animals also being used for to see that movement of tracking movement and their feeding, breeding, all these things are seen. But this wind, I think, would be crucial parameter. Bathymetry, no doubt, directly, no doubt, it is indirectly, it is related. What asked by question, recent question asked. So it is indirectly because it will tell, because the type of fish is available, and it is also linked to bathymetry like pelagic, demersal, all these things, bottom trawler, and even long line. All these things will be used by this fishermen, so that bathymetry is a indirect parameter to say that because no doubt in coastal, in shallow water, in continental self water, you will have more means birds movement will be there, because we also participated in many vessel movements and vessel means cruises. So bathymetry indirectly will tell, but exactly you can't say that more or more birds will be means will be there observed. That exploration is needed. That is a
Yeah.
topic of research.
That's true, sir. That's true. Whatever you said is you pretty much agree with you. The thing is that as you said, wind. You're referring to the wind in the beginning. So wind, as you say, in the sense in the case of Bay of Bengal. So even though we have these cyclones, so the cyclones are pretty much frequent in Bay of Bengal. So as you say, probably you might have thought that wind is a very major factor in this.
Yeah.
So what happens is, like, basically in the case of seabirds, as well as when you're linking with that fish shoals, the thing is that in Bay of Bengal you don't have much mixing happening between the surface and deep waters. So
Yeah, yeah.
when this mixing happens, and the nutrients usually upwell. So the upwelling actually is not pretty much of intense in BOB because of this strong surface, layer over there. Even though you have severe bashing of the cyclonic storms every now and then, actually because of the continuous runoff from various river output, input. So you have this strong stratified layer on the BOB, Bay of Bengal.
No, actually
So,
not exactly. I am telling this seabirds movement pattern, not
yeah, yeah,
exactly
yeah.
this, not
So
ocean
I'm
features
just
related. Yeah, yeah,
yes, sir. I'm just relating to that, to the
yeah,
oceanography feature, which I'm telling you, because
yeah,
wind is a major factor in actually bringing out the nutrients on the top.
yeah,
So because
yeah,
of the nutrients, actually these fish happen to be there, and
yeah,
this
yeah, right, right.
and
Upwelling, it is more
yeah, and
related to
fish
upwelling.
depends on plankton, so everything is like related here. So when
Yeah,
you're referring to wind, so what I found during my study, actually wind seemed to be not pretty much of a strong indicator of this seabird distribution. So when I have done an exploratory, since it's basically mine is an exploratory study, so we at a higher level. So when I'm generating a model, so that wind factor also would be a very will play a crucial role in that. But for that we need more data set, and a long-term data set would really help in giving a good answer to that.
no,
And
actually,
coming
I'm
to
telling
the
they have to track. Wind actually probably decides the seabirds track also. Like in
yes, sir. Yeah, but you're telling about the surface, probably you're telling about the surface winds, no? So
atmospheric winds.
atmospheric winds, yeah, atmospheric winds as such, like that one, that is a indicator also, but it depends on the seasonal, actually there are seasonal patterns. So
Yeah,
for
yeah, right,
that you
right.
need to generate long-term data sets, so to know, since
Okay,
I could only
thank
yeah.
you,
Thank you,
Dr.
sir.
Ravi, because of
Yeah,
yes,
Yeah, yeah.
time on
Thank you, thank you.
and
thank you
one
very
more
much, sir.
question we'll
Thank you.
allow from Dr. Rafi. You want to ask the final question?
Yes, sir.
Yeah, Dr. Rafi.
Is it audible, sir?
Yeah,
Yes,
yeah.
sir. Yes, sir. Audible.
So I have a basic question to Dr. Sarangi and Dr. Deepak Sabal, because both of you are marine biologists. Can you tell in brief how your way from marine biology to become a remote sensing scientist to Dr. Sarangi, and also to Deepak Samuel from a basic marine biologist to become a coastal zone management expert? This will be a boon for our students, please.
Hello.
Yes.
Good evening, Dr. Rafi.
Yes.
Okay, okay. Let me complete means answer first. Then I think I will briefly, very shortly, I will answer, and then Dr. Deepak can answer. So as we know, you are also from same university. I was your senior. So that time, so because remote sensing, I entered into this field then, which is able to map, monitor, manage these coastal ocean resources in very precisely and synoptically, as you know. Like I mentioned earlier, you might be attending just half an hour back regarding these satellite channels, sensors, and all these detectors, everything available in Indian level and in nationally, globally also, like European Space Agency, NASA, and different space agency. They have different sensors, like from microwave means that hyperspectral to optical, then UV optical, then infrared, all type of infrared thermal channels, then microwave radar, all these are there. So that plays a great role to have this mapping, monitoring the resources. So being a marine resource student, as we thought that I thought that this is a great opportunity to initiate a career in ISRO, and somehow we with Almighty's grace able to get sufficient work, good team, and good organization to have variety of projects, to have different means kind of projects and colleagues and students to monitor our resources and to map and to have different kind of understanding of Bay of Bengal Arabian Sea processes, then parameters, features, then how it can be linked to this from primary, secondary, tertiary producers like phytoplankton, zooplankton, fishes, then mammals, now birds. Recently, just now, last speaker, Dr. Ravichandra, he spoke about the birds. So that way, it is very, very means useful platform to have this marine. As you know, is 2/3 of our planet. So no doubt it is always, it will be challenging and very, very diverse subject. We have linked to atmosphere, ocean, land in regional to global scale. So
Okay.
you know that,
Thank you, Dr.
so that way, yeah, that way it is very important. Now I hand over to Dr. Deepak can tell about this, his
Deepak,
expertise.
can you wait? We can answer towards the end because we have 6 more questions.
Absolutely, no problem. Please go ahead. Please go ahead. Absolutely.
Okay, now we have another presenter, Dr. Ravinish, who is working as a postdoctoral fellow in the Department of Aquatic Biology and Fisheries at University of Kerala. He is working on mollusks for a long time and a very good field biologist and taxonomist. And his expertise in field work actually helped him for selecting in the Hawaii Biodiversity Assessment Program as well. Dr. Ravinish.
Hello?
Dr. Ravinish, yeah.
Yeah. Can I start my presentation?
Yeah.
Yeah. Hi, I'm Ravinish. I'm working on postdoctoral fellow, Department of Aquatic Biology and Fisheries. And
please share your screen. You're not shared your screen yet, Ravinish.
You share your screen.
Okay. Okay.
No, not. Yeah. Okay. Make it full screen. Okay.
Yeah.
Okay, go ahead, please.
Yeah. As you are aware, mollusca happens. In recent studies, estimates the extant species of molluscan diversity is around 45,000 to 50,000 species. That mollusca phyla is divided into 8 living classes. Two extinct ones. The database of Indo-Pacific molluscan provides the nomenclatural distribution and ecological information on 28,350 species in Indo-Pacific region. In India, molluscan diversity studies mainly conducted on coral reef ecosystem-based studies. The majority surveys were conducted as a part of comparison of data for general biodiversity databases and all phyla studies. In India, around 5 phyla were recorded. That gastropoda is most dominant, more than 2,000 species recorded. That bivalves are the second largest class, around 750 species, 750 species were recorded. Then other classes like Capopoda, Cephalopoda and Polyplecopoda very least amount, approximately 100 species were recorded from various parts of Indian region. As part of my PhD work, 500 species of molluscan were collected from various islands of Lakshadweep, and so far I'm published 33 research papers and 5 book chapters, and visited 3 overseas. Three overseas like Malaysia, Singapore, and Hawaii. That already participated in diversity program in Hawaii. Now my achievements means now in our collection more than 1,300 species were collected from various parts of mainland coast of India and Lakshadweep. It is 23 species are new to science, more than 100 new records from new to India. Now we are working on field guide about molluscans of Kalpapannar and surrounding seas. It is around 1,000 species field guide with collaboration with Dr. Patterson. Sir. These are the photographic and pattern of our book. It is fully colorful guide. We are making colorful pictorial guide. That in molluscan area is these are the gap areas. Normally, most we need more in-depth studies like ecosystem based surveys are required, and in rocky shores, coral reefs, lagoons, mudflat, sandy beaches, and estuaries and deep sea, especially deep sea and seamount region. No one will study it. That is also need to more work. Then my future plan is I like to make one compendium of marine mollusca of India, and the next one I like to explore that poorly documented deep sea mollusk. And another thing, in India, India is the molluscan part of India is still poorly known because that not everything endemic to India. Maybe same species is closely related species are living on that Western Indian Ocean and Eastern Indian Ocean, Pacific region, that area also. So I like to work biogeography and integrated taxonomy also. That this COVID time, I am playing with my data and specimen, and I like to take very good photographs. Same photographs already taken from my own. Thank you. Yes. Hello. Hello, sir.
Deepak, any questions?
Just want to compliment Ravinesh, sir. I think another excellent budding. So one question to me is that remember we will discuss more about micromolus. So I think that should also go into the agenda because we have very few people working, and probably it's time that we trained the next generation people also. Now we should pass on our talents and our approach to taxonomy to younger generation. That's it.
Yeah, now our collection, we are identified more than 70 micromolluscan also. So we are
actually, micromollusc is one area where somebody has to invest his or her lifetime to become a trained taxonomist. Yeah, Dinesh, Dr. Dinesh.
Yeah, I'm audible.
Yeah.
Ravinesh, thanks for the excellent presentation, and it clearly shows how much time you have invested to collect all these, samples, and you roam around all the places. So I'm really wondering, like, you have collected so many samples, and I hope you preserved it for DNA extraction and all those stuff. I think you really mentioned it, like you're doing some molecular work. So do you have any idea to work on population connectivity, in one particular, for example, like endangered species, how it is connected to each other, how it is migrating from one place to other place? Is there any study in your mind, or is there any studies already being carried out there?
No, currently no. I like to work on feature.
Oh, you like to work on feature? Okay. So I hope you keep all the samples in your hand because collecting samples is not that easy. Thanks, Ravinesh. Thank you.
Thank you, Dr. Ravanish. We'll move on to the next presenter, Dr. Chandran Datanaj, who is working as a marine biologist at the Gulf of Kutch Marine National Park and Sanctuary, Gujarat. And he's an octocoral taxonomist and did his PhD on octocorals. And currently also working on reef restoration. Dr. Chandran? Yeah.
Am I audible, sir?
Yes.
Shall I start?
Yeah, you make it full screen and start. Yeah.
Already it's full screen only, I think.
Okay. Okay.
Is it not?
Yeah. It's fine.
Yes. So, good evening, everyone. I'm Dr. Chandran, presently working as a marine biologist in the Marine National Park and Sanctuary, Jamnagar, comes under the Gujarat Forest Department. Presently, my role in the MIP is to assist the Marine National Park and Sanctuary Authority on implementing conservation activities and interventions. Basically, I'm an octocoral taxonomist. Did my PhD on the taxonomy and biodiversity of soft corals. Octocorals are a fascinating animal group. They are commonly seen in and around the coral reef. Until the recent past, scientists were believed that the octocorals are not playing any role on the coral reef while coral reef construction and all. But with the recent studies revealed that octocorals are playing some crucial role on the coral reef ecology, trophodynamic, even the construction of coral reef as well. Octocorals is a collective term to mention the corals belonging to the subclass Octocoralia of the class Anthozoa phylum Nidaria. So this group includes softy and flexible corals. Flexibly, a variety of corals include blue corals, soft corals, cauliflower corals, horny corals, sea fans, and so on. So, as of the present data, the octocoral presently comprises of over 3,649 nominal species, divisible under 45 families, and 334 genera. In the global scenario, there is a growing interest on octocorals as they have been proven to have a magnificent source of novel molecules and interesting metabolites. So this is the classification taxonomy of the octocorals. The order Chilioporaceae, order Alcinoaceae, and the order Pennapilaceae all includes this octocorals. So as far as its diversity is concerned, octocorals are very rich in diversity than hard corals even. So in some reefs you can see more diverse soft coral than hard corals. So the octocorals can be seen from the pole to tropical. They can be seen from the intertidal to abyssal depth even. So they are very much diverse. More than 70% of the octocorals are reported from the Indo-Pacific only, especially the families Zenidae, Neptidae, and Alcinidae are the very common species available in the Indo-Pacific reef regions. Then for the Indian scenarios, the studies on the octocorals have been was started since in the year 1905 during the British period. Still, we have reported only 253 species. Even in fact, ZSI has a lot. It's the largest collection of octocoral type specimens in the world. So perhaps the difficulties in the taxonomical identification and the inavailability of identification keys for many families and the scarcity of taxonomists in India or might be the reasons for the less report from India. So, with regard to the diversity, so far 253 species have been reported, comes under 21 families. Most diverse is Andaman Nicobar reefs, it's 190 species, followed by Gulf of Mannar 47 and Lakshadweep 40. So this very less number is not because of the less diversity, it's due to the less effort invested on the exploring diversity. So I've been collecting, exploring the octocoral diversity along the Gulf of Kutch also. I'm also expertising on coral transplantation in the Gulf of Kutch. Under the supervision of Dr. Satyanarayana, I had a chance of leading a team who was transplanted more than 1,000 square meter degraded reefs in the Gulf of Kutch, three spatially distinct region along the Gulf of Kutch Marine National Park. So it was a successful attempt to restore in such very high sediment reefs. So we have used native species as well as transplant branching coral species from Gulf of Mannar. So we could reintroduce the branching coral species once again in the Gulf of Kutch after more than 10,000 years. So we have developed strategies on coral transplantation in sedimented current influence regions as well. So these are some of the coral transplanted native species and branching coral species seen on our degraded reefs region in the Gulf of Kutch. So interestingly, a 7-centimeter branching coral fragments collected from Gulf of Mannar have grown up to 41-centimeter diameter colony within 2 years. So these are the nursery site. So recently I have been expertised on the advanced way of coral transplantation called biorock coral reef restoration. That involve use of some low voltage electricity to promote the coral growth and restoration of the degraded reef regions. So this technology is a basic electrolysis process only, using some low voltage electricity from some solar panel or some electric source from the land can be used for promoting and restoring coral reefs. So we have been trained by the Dr. Thomas Gorio and Dr. Satyanarayana sir, the experts on this group. And presently, Gulf of Kutch has 2 biorock reefs. They are working very well. This is the accretion rate within 1 month. So these are the coral transplants on the biorock reefs. So interestingly, within 2 weeks, they have started overgrowing the tide material and started doing incrustation growth. So the data also reveals its success. So more than 1.77 centimeter per month linear extension growth was recorded in a biorock reef. Then accretion rate also very high. So it was recorded up to 0.1 millimeter per day. So it has been proven that biorock is a suitable technology for restoring coral reef in the Gulf of Kutch and other reefs of the country. So the results are there is a assemblage of fishes and other biological ecological also proven that this biorock has been working properly. So this was the team we have together. We have installed that biorock. You can see the director of ZSI, Dr. Satyanarayana sir, Dr. Thomas Gorio, and all forest officials. So recently, my popular writing on what's that? entitled Save Coral Reefs, the Paradise on Earth, has been awarded 90. So further research, further way forward in the post-COVID-19 era. So I would suggest, as all mentioned, the collaboration with the institution working in similar objectives, sharing the samples for every, say for example, if the soft coral, the people working on molecular studies, working on chemical aspects, compound studies, can be collaborated with the field staff taxonomies. Like that, we can move forward in the COVID-19 eras. And thank you, sir. Thanks a lot for giving me the opportunity. Thanks a lot for the organizers. Thank you.
Thank you, Dr. Chandran. And it's so open for discussion. Any specific questions to Chandran? Now, my question to Dr. Chandran is that, see this biorock technology, when you use electric current in a mild dose, will that have some kind of repercussion on the reproductive success of this organism, whether that biology part has been studied or not?
So actually, the biorock reef has the methodology has been proven to have more growth rate and even every aspects. Basically, they are promoting the metabolism of the coral, so in such a way that they can have more calcification, more growth, more feeding capacity, everything. So I don't think that they will affect their breeding.
Okay. Dinesh, you want to ask something?
Yeah, so thanks, Chandran, for the great presentation. Like, I know doing transplantation in the high sediment area, how tough it is. Like,
yes.
You've done a good job. But I have a few questions. I mean, not few questions, one or two questions. Before we take, this is the question raised by many researchers in India. Before we take the corals from Gulf of Mannar to Gulf of Kerch, like, I'm curious to know what are the parameters you really checked in. I mean, I know that it's success really well. And, what is my worry is, in case, if the particular species is weedy species, or if the particular species no longer reproduce as Professor mentioned, so what will be the impact? Like,
yeah, it's a right question. Thing is that before selecting the species for transplantation, a detailed study on the fossilized corals have been carried out. So based on that, we have identified what are the species which were covered the Gulf of Kerch reef before. So based on that only, we have selected the branchicoral species. And this, I think I have answered.
Okay, so one more interesting stuff we could look at into it is that, okay, so you have taken corals from Gulf of Mannar to Gulf of Kerch. So based on the work what I'm doing right now in coral symbionts, what is telling me is that the symbiont what is existing in Gulf of Mannar is also existing in Gulf of Kerch too, because when you do transplant it, initially the coral partially bleach, then it started to recover, right? So those are the very interesting, sides to look into it. And
yes.
The collaboration with those aspects definitely fruitful.
Yeah. And so we also have proven a lot of observation in such a way that this Gulf of Kerch as as someone asked, if we have, I would say that we have resilient corals, super corals in Gulf of Kerch. So. So they are very tolerant species. Of course, there is no impact of mass coral bleaching and all in the Gulf of Kerch much. So it's a really a good place to restore, transplant and conserve in the future point of view.
So can I ask one more last question?
Yeah, please. Quickly.
So the last question is, regarding the VOYO rock. That is one of the interesting area scientists are proposing. At the same time, we should be more careful that there are so many countries not ready to accept it. Including in USA, I talked with many, many researchers. They are literally scared about it because what exactly going on in, as sir mentioned, what is exactly going on in the biology part. In case, for example, in case if some mutation happens in the coral organism, what will happen? If we put a rod, some mutations happen. If some species started to grow really faster than what we expected, then those things concerned really need to, really need to know. Okay, when you give the electricity, what happened to the biology? How it is constructing the reef faster? How it is accumulating the calcium really faster, and how it is attracting recruits?
Yeah, basically this biorock, the electricity given to the biorock will promote the metabolism with ATP cycle and all. So they will be promoting the metabolism so that all their biological activity will be speed up, including calcification. So that might be the reason they grow faster. In fact, Gulf of Perch is such a reef where we have to do a faster way of coral restoration efforts is needed. So rather than going for coral transplantation, rather than the slower process, so we should check its the efficiency of biorock on the Gulf of Perch is really need of the hour. So for the changing climate and other things, other points of view.
Okay, thank you, Dr. Chandran. Thank you, Dr. Dinesh, for the questions. And it opens up another interesting field of study, probably involving some kind of gene flows as well, genetic flows and between the populations, and interesting.
Okay.
Yeah,
Thank you.
Thank you, Dr. Chandran. And next one to speak is Dr. Anish, who is working as a UGC postdoctoral, Kothari postdoctoral fellow, and his field of research is taxonomy, biology, and taxonomy and biology of parasitic crustaceans. He is a person who insists that the publication should be done only in the highly reputed and impactful research journals. Dr. Anish. Dr. Anish?
Hello, sir.
Yeah.
Hello. Very good evening to all. I'm very happy to see all marine biologists here. Today we are celebrating World Ocean Day. In this occasion, I would like to share my area of research, and I would like to introduce the area.
Yeah, your video is not there.
Sir, hello.
Your video is not there.
Sir, actually, I have some problem with the internet. That's why.
Okay. Right. Okay. So make it a full screen and then present here.
In past 1 decade, hello.
Yeah, you can speak.
Audible. Hello.
Yeah. Speak, please. Yeah.
Hello.
Yeah.
Sir?
You can speak. Yeah.
Hello. Okay. For the past 1 decade, working on various aspects of parasitic crustaceans, especially biology of parasitic crustaceans infesting the aquatic fauna.
Hello. Hello, Anish. Anish, you are not audible.
Hello.
Hello.
Hello. Sir,
Not clear. You're
can you hear me? Hello.
not very clear.
Hello. Sir, can you hear me?
The sound is not clear, Anish.
Okay.
Now we'll come back to you after some time. It's okay. Hello. Anish, we will come back to you. Okay. Anish, hello. Can you hear?
Hello, sir.
No, your audio is not that good.
Not
No, your sound is not clear.
Hello.
And I think there is some problem with the connectivity. You have. Now what you can do is you can wait and hello.
Hi. Hello.
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 Hello, Anish. Hello.
Sir, there have some net problem. He will present later.
Hello, Anish.
Dr. Biju, maybe we can, take the next one, and somebody can call Anish and tell him that he can join at the, later stage.
Yeah, I was call him. There is some net problem. He will join later. We can move to next person. Possible.
Anish? Okay, right. Sorry, some issues with his internet, and we'll just move on to the next presenter, Ramvilas Ghosh. And Ramvilas is a PhD scholar at the Kerala University of Fisheries and Ocean Sciences in Cochin, working on the diversity, distribution, and evolution of gorgonians of Indian waters. Ramvilas?
Sir, am I audible?
Yes.
So I'm a researcher from Kerala University of Fisheries and Ocean Studies, and my research topic is on gorgonians, which is towards bridging the Linnaean, Wallacean, and Darwinian shortfall in Indian gorgonian fauna. So basically, gorgonians come under the order Alcinaceae, which is again divided into three suborders: Catcaxonia, Prolaxonia, and Scledaxonia. So gorgonians basically come under these three suborders. So why gorgonians? See, there are many ecological importance of gorgonians. For example, they form large ecosystem services in, especially in the deeper regions. And there is a global hunt for prostaglandins from gorgonians. And there is a misconception that corallium species, which is known as the precious red coral, which fetches some around several $1,000 for a single kilogram. Many people think it as a hard coral, but actually corallium is a gorgonian. So, and gorgonian is also indicator of several environmental factors like sedimentation, temperature variations, just like corals. And there is also some aesthetic values of studying these charismatic organisms. And when it comes to the Indo-Pacific region, even though, as Dr. Chandran has quoted, even though this is a region of high diversity, this is a region which is known for its minimal knowledge on gorgonians. So this is my aim, that is to improve our understanding of diversity, distribution, and evolutionary biogeography of gorgonians in India. So the objective is to determine the diversity, distribution, and gorgonians of Indian coast, to understand the molecular phylogenetics of genus Gensela of the family Elisilidae, and to determine the phylogeography of Gensela, which is again the family Elisilidae occurring in India. So as I already told you, the justification is this, that the Indo-Pacific region is deemed as the region of minimal knowledge, particularly in the case of gorgonians, highlighting the scattered and poor knowledge, especially in their taxonomy and diversity. So these are the basic research questions that I'm trying to answer. What is the current status of gorgonian diversity in India? What is the evolutionary history or lineage of genus Gensela, which is also known as sea webs? Are the populations of webs occurring in the Indian mainland and islands genetically connected? If yes, what is the direction of gene flow? Or otherwise, the reasons for isolation? So this is the background for my chapter 1. Based on my gorgonian collections, the diversity is different in different ecosystems. For example, Lakshadweep, Gulf of Mannar, which is a fringing reef, Goa, Mumbai. So everywhere we see there's a stark contrast in gorgonian species diversity. There are some commonly occurring species, which is Gensela. Other than that, we can see a stark difference in the species diversity. The chapter envisions to highlight these exceptional species diversity changes with change in ecosystems, which will also add as a baseline data on the governing diversity in India. So these are some of the differences that we can see in different ecosystems of India. And the next chapter 2, the CB for Jansila species, which is a common presence in the eastern and western India and Lakshadweep. So this is one reason I thought about focusing on this particular genus, because as far as my collections are concerned, this species is available, or this CWIP is available throughout the coast and islands of India, Lakshadweep and Andaman. And the genus Jansila currently, according to the current taxonomy, there are 4 species and has an Indo-West Pacific and Red Sea distribution. So in a recent publication by biologists in 2014, the authors have pointed out that the level of divergence within the Jansila is high, that there is a chance of cryptic speciation within the species, within the genus. So they suggest that at least 3 new genus can be found within this particular genus. So this is one motive to take this genus as my target species. So this is the evolutionary study by Biolwich. And next, chapter 3 is the population genetics of Jansila. Since the availability of Jansila samples are there from coastal regions of India, including Lakshadweep and the mainland. And so far, I have one publication
very good.
which I have published. So regarding the COVID research, one thing I am there are many constraints that I am facing now. For example, now I have to change the chapter 3 population genetics due to funding crisis. And I was planning to complete my field by this June, but I already missed 2 months of my field trip. So basically, what I'm trying to do is I'm going to work on what I have, whatever the samples I have, and these are some of the constraints that I'm facing. And regarding the collaboration, I'm actually right now collaborating with many of the international experts in my research, and I'm also in touch with other guys who are working in soft corals and sea fans in India, like Dr. Chandran. Actually, we are communicating and we are actually sharing samples for molecular studies. So yeah, these are my work progress so far.
Chandran, Chandran, can you just mute your audio, please?
Okay.
Sorry. Hello, sorry. Any specific questions to Ramvilas? Dinesh, please. Yeah.
Thanks for the excellent presentation, course. And you are collecting so many samples from different geographical locations. That's more curious. So in terms of hard corals, like we know that when the corals are reproducing and how it is, in Gulf of Mannar, we have recorded it, it's reproducing in March. But how about the reproduction, reproductive biology in the Gorgonians? Is there any report in abroad, at least, like how it is? It's the same hard corals or?
Yes, sir. it's I think it's the same as the hard corals. There are both sexual and asexual reproduction methods. And for example, some are mouth brooders, and some are broadcasters. It's the same as, I think it's the same as hard corals. There are many studies abroad. When it comes to India, there are no such studies. And I have also seen these sea whips, which is Gensila. They have this capacity to drop off their branches. I mean, there are just they look like whips. They can just drop off their fragments, and they'll drop to the ocean floor, and from there they will arise into new colonies. So these kinds of reproductive methods are also there.
Thanks, Ram. And we have 3 more speakers, and then Deepak, you want to ask something? Deepak, you want to ask?
Just a quick question. Yes, sir, just a quick question. Ramvilas, nice presentation. Just want to know, is Juncilla the only species we have in the country, or do we have? You said there are kind of 3 possibility of 3 species available. But in your experience, what do you think? Is Juncilla the only species? That's my first question. And the second question is that did you find any ectosymbionts, like or kind of other symbiotic species on growing on them?
Yes, sir. Yes, sir. Thank you, sir. Actually, I think from my experience, I think there must be differences. I think there must be at least more than 1 or 2 species. I'm working on it. Currently, I'm working on it with one of the international experts, and we are working on this taxonomy. I think soon I will be able to solve it. And yes, there are many ectosymbionts. I have seen whenever I get these gorgonian samples, there are brittle stars attached to them. Each and every time I get them, there is brittle stars, and sometimes you'll get crabs, some small crabs, and I mean, some of them are, I don't know what they're I can't even identify.
Basically, I asked this question mainly because there is one particular bivalve, Tyria winged oyster, which is very common. So that's what I wanted to.
Okay, thank you, Ram, and we'll now move on to Shalu, who is doing her PhD in Kerala University of Fisheries and Ocean Studies in Cochin, and she works with the syngnaathids, which include the seahorses and pipefishes. Shalu.
Is it audible, sir?
Yes.
Yeah, I just move on to my slides. Can you see my slides? Hello?
Yeah, go ahead, Shalu.
So I'm Shalu. I'm doing PhD in Kufos. So my work is taxonomy and conservation of syngnathid fishes of India. So I'm working on seahorse and pipefishes. So everyone know what is sygnathids. It has, and the family has seahorse, pipefish, pipe horses, sea dragon, and it, as everyone know, it is a charismatic species, but also it is a conservation concern. They are also important in ecological, economic, and medicinal importance. So moving to my work, so I'm working because syngnathid fishes are poorly studied group which are threatened due to overexploitation in India. So what is my, so how many species of syngnathid do occur along Indian coast? So there are studies about seahorses, but there are few studies about pipefishes. So I'm working on both the species to know the diversity in Indian coast. Then I'm moving on to know about the exploitation rate, because these species are highly highly exploited in India. So I want to know what's their growth, their mortality, and exploitation rate in some of the areas, to understand what's the current exploitation rate in India. And then I move on to know about the genetic variations in Cycnatha species from east and west coast of India. So I saw there are morphological variations in seahorses, same species from different coasts. So I'm working on it, but when working on the genetic variation, the genetic variation is very few. So I'm actually working on this aspect. Then next, coming down to the last one, what are the existing policies, and is that the managing measures effective in conserving methods? So this I'm actually saying a brief idea of what I'm doing. So these are species that are occur in India. So as you see that 70% of the species are vulnerable. 70% of species are in vulnerable condition, based on IUCN Red List. And about the pipe fishes, most of the species are data deficient or least conserved. These are some of the paper cuttings that you have seen about the smuggling, these species are illegal, are under Schedule 1 under Wildlife Protection Act. Even though these are scheduled species, both seahorse and pipe fishes, still the illegal trade is continuing. So I'm working on that aspect, and working on the community level, that how many people Protection Act, and what about their perception, their knowledge about seahorse methods, and what is their attitude towards conserving this organism. So is that they are dependent on the species, or like that. So I'm doing a survey on that, and I'm aware people about, because most of when I did the survey, I feel that most of the people are aware of seahorses are under protection and it is illegal, but none of them knows about pie fishes are illegal to catch. So I'm doing, and some of most of the East Coast people are more aware compared to the West Coast. So we are doing some awareness to know about how to, because conservation need to be come from the community level. So we are doing some work to aware the people about Wildlife Protection Act and about species. So this is a brief idea of my work. I'm not getting into too much deeper in my work. So I think it's what conservation turned the light in their favor. So that's about my presentation. Hello. Hello. Is it audible? Hello.
Yeah, you're audible, Shalu. Just wait for Viju sir to respond. You're audible. No problem.
Ajita, you are audible and you can
Yeah.
ask. Yeah.
Yeah. I thought I'm just talking to, because yeah.
Ajita, you have some question? Hello. Ajita.
Hello.
Hello.
No, sir.
Yeah.
No, sir. No. Nothing.
Okay. Any panelists? Any question to Shalu?
Not a question. I just wanted to tell her that on the similar line, there are already 2 theses from NIO, and there are some couple of papers are already published, both on the molecular and morphological, as well as both pipe fishes and seahorse, as well as from the Annamalai University. It will be good if she can refer to those publications, and that will help her to modify and take the lead from that. Thank you.
So my adding to what Bhavan sir has told.
Actually, I'm communicating with them also.
Yeah, Deepak.
Sir, just adding to what Bhavan sir has told, the illegal trade route also, that's again a big concern. And the Wildlife Crime Control Bureau and also WWF, they are trying a lot to kind of curb this. But it's a very challenging work he has taken. I think this work will definitely help in bring out some policy. So wish you all the very best.
Yeah, it's a challenging work. All the best, Shalu, for the work. And now we have two more presentations. Quickly, we'll go through that. One is Al-Badusha. Al-Badusha is around. Al-Badusha is the research assistant with the Oceans and Coasts Program, Nature Conservation Foundation in Mysore, working with the sea turtles, specifically sea turtle seagrass interaction in Lakshadweep. Al-Badusha.
Yeah, thank you, sir. Am I audible? Am I audible, sir?
Yeah.
So, 1 second. 1 second, just. Yeah. So I'm working with the sea turtles interactions of Lakshadweep. So sea turtle populations are decreasing all over the world, but Lakshadweep is one of the very few places in the world which has a high density population of green turtles. So before 1972, people used to kill sea turtles for their meat. Just a second. Yeah. So people used to kill sea turtles for the meat, which used as a waterproofing agent for their country boats. After the enactment of Animal Protection Act, people stopped killing turtles, and thankfully, a lot of conservation activities are happening around the globe, and which help the sea turtle to increase their number. And the sharks are a targeted fisheries in the world, and the tiger sharks is one of the main predators of green turtles, which their numbers have decreased. So the number of green turtles have increased in Lakshadweep. So we are monitoring the sea turtle high density of feeding population of green turtles in Lakshadweep since 2005. So what happens in Lakshadweep is turtle comes as a really big group to an island, which eats all the seagrass meadows, and they've moved to another island, which makes the sea turtle have a really big conflict with the fishermen in Lakshadweep. The fishermen in Lakshadweep are highly dependent on their meadows, the seagrass meadows, because they get their bait fish for tuna catch. So Lakshadweep people are mostly fishermen who are highly dependent on the seagrass meadows for their bait fish. So they use live bait for the tuna catching. Plus the reef fisheries is really big thing in Lakshadweep. So we started observing sea turtles and seagrass interactions in Lakshadweep since 2005. So what we're seeing now in Lakshadweep is, like I said, the turtles are a really big group which comes to an island, which started grazing on the island, and it eat all the seagrass meadows, and which it completely vanished the seagrass meadows, and they moved to another island wherever the seagrass is present. So what we're seeing in Lakshadweep is turtle come as a really big group, and there are a lot of interactions which are happening with the seagrasses. So they prefer Thalassia seagrass, which is a late successional species or a stable species, which is long-lived. So turtle mainly focus on Thalassia first, because it's called turtle grass. So once the Thalassia is exhausted, then they move to another succession level, which is a Cybodosia, another species which is kind of short-lived when compared to Thalassia. So we started observing change in preference of seagrasses in turtles. First they choose Thalassia, then they move to another seagrasses. Then they start, once it's exhausted, they start eating algae plus whatever seagrass is available.
So what's happening here in the map, I'm showing just an example of an island, which is Agatti in Lakshadweep. So 2011, you can see a big dark circle, which is the highest density population of seagrass which we've observed in 2011 in Agatti. So they started grazing. So what we're seeing in the seagrass meadows is Thalassia seagrass has been exhausted completely. Then there's a species shift of seagrass meadows. Then there is first it was a Thalassia dominated species meadows. Then it will become a Cymodosia, which is a comparatively low living, short living species. And there's a barren stage. You can see in 2019 in Agatti, the turtles have eaten all the seagrasses, and then they moved to another island, which is Kalpeni. And in Kalpeni 2019, they have eaten all the seagrasses. They have moved to another island. So what we're seeing now, after exhausting all the seagrass meadows, what we're seeing now is they've changed their group behavior. So earlier they were moving as a group, and now they've changed their group behavior, and now they started moving independently. They started exploring other islands. Earlier we were seeing high density peaks in different islands. So if you're seeing a really high density in one island, there may not be a big density in other islands. Now what we're seeing is they're evenly distributed in all the islands. So they've changed their group behavior into independent behavior. And the seagrass, the way the seagrass are responding is they're shifting from the long living species to comparatively short living species. And you can, as you're seeing into 2019 in this island map, there's a barren stage where there is no seagrass at all. And in 2020, we've started seeing a slight recovery of seagrass, which is very short living species like Halodula and Halophila, which are comparatively very short living species and pioneer species of seagrass meadows. And it's an ongoing study. This is what we're seeing in Lakshadweep. Thanks. Thank you. That's it.
Dr. Sarangi, you're asking something?
Yeah. So yeah, very nice presentation. And this work actually, this turtles, is it only green turtle or any other kind of turtle they used to do the grazing there on seagrass meadows? And this, is there any satellite map? Have you referred anything to this like classification and this seagrass community zonation?
So it's mainly green turtles in Lakshadweep, and there we can see olive redleys and olive redleys as well.
Yeah.
But we haven't done any satellite images. We're just directly observing every year since 2005.
Okay, and actually these turtles, they are grazing a lot of seagrass meadows.
Yeah.
So how easy, our concern is to conserve the turtles as well as seagrass. Both are of concern. Then how do balance both the things? What is your opinion?
So we think about turtle as a part of the system. So we have to consider both seagrass and sea turtles. Now what we're thinking is, so if many people are conserving turtles, but in Lakshadweep, if we talk to the fishermen, turtle is a pest for them because it's coming to their island and it's eating all the seagrass meadows and then moving to another island. So over a period of time, they're declining the whole lot of reef fishes which are used as a bait fish or a food. So if you're considering it as a system, so we need both seagrass and sea turtles. So what we're doing now is we're trying to conserve both. Turtle, we're talking to people. Earlier, people used to kill turtles in Lakshadweep. They used to burn turtles when they come to the shore for nesting, and they used to chop their flippers, which is very easy to catch the turtle. They'll catch the turtle, they chop the flippers. Now, so we've started this outreach and awareness, and people started thinking the problem with the fishermen is the availability of the less availability of the fish, not the presence or absence of sea turtles. So if they're getting proper fish, then there's no problem there. So now we started conserving seagrasses. So we started putting exclosures for seagrasses in several islands, and we started this outreach program, and we started telling people that Lakshadweep is supporting a global population of a global species, which is and
And the total come round the year or any particular season?
they'll be there for round the year, all the seasons.
Okay.
Thank you, Al Badshah.
Yeah, thank you.
Thank you so much.
Thank you for the nice presentation and nice work. And final presentation will be by Mr. Riaz. So he's working in University of Kerala, Department of Aquatic Biology, on the taxonomy of jellyfish, and also he's working on the venom proteomics. Riaz.
Is it visible?
No, it is not. Share the screen.
Sorry. Yeah. Okay.
Yeah.
Okay, sir.
Yeah. You make it full screen and present. Yeah. Okay.
Riaz. I am a research scholar in the Department of Aquatic Biology and Fisheries, University of Kerala. I am doing research under the guidance of Professor A. Bijukumar sir. My topic of my research is taxonomy of nertic jellyfishes and studies on jellyfish bloom from the Kerala coast. We know jellyfishes belongs to the phylum Cnidaria, class Scyphosova, Cubosova, and Hydrosova. Among this, Scyphosova and Cubosova is considered the large jellyfishes. So we are concentrated on Scyphosova and Cubosova. In this ocean day, we cannot ignore the functions and the to our ocean ecosystem. So I am moving to the importance of jellyfishes. Jellyfishes play a key role in the food web of marine ecosystem. It provides food source for the bottom dwelling corals and solitary bottom dwelling is one of the prey of leatherback turtle and some other organism like marine seabirds and mola, the sunfish, also a good predator of jellyfishes. Jellyfish have a significant role in the aquatic biogeochemical cycle. It helps to mix throughout the carbon mix throughout the ocean. It is an important item in the aquarium trade due to the beautiful coloration and the pulsating movement. It is used as a aquarium keeping. In other ways, jellyfish is used as food. In some of the Southeast Asian countries, especially China, Japan, and Korea, they use jellyfish as food. In some state, Andhra Pradesh, Gujarat, and Tamil Nadu also exporting jellyfishes to the Southeast Asian countries. In addition to that, some bioactive compounds also isolated from jellyfishes from the tentacles, that is the nematocysts. They contain some venom, the venom proteins are good for some medical treatment. In addition to that, some collagens also good for some wound healing treatments or medical treatments also. That's the important aspects related with the jellyfishes. Now I am going to the slide. Move. Okay, sorry. Now we are moving to about the jellyfish population. Nowadays, for the past few decades, jellyfish population increase in our coastal lakes. It creates severe menace for the fishery activities. So it's a loss of fishing days because it's a menace for the fishery activities. It is clogging to fishing nets, and it's stinging to the fishermen during its fishing operation. In India, there is no good study or in-depth study about jellyfish blooms, dynamics, and the diversity of jellyfish. In addition to that, we don't know about the envenomation, which are the species causing venomation, envenomation to humans. So in this situation, we are trying to take this topic for our further research. I am working in this field to study the taxonomy of jellyfishes of Kerala coast using morpho and molecular approaches to study, to document the jellyfish blooms and the dynamic along Kerala coast, to document the reason, possible reason behind jellyfish bloom along the Kerala coast. I collected the specimen from beach scene and prowl by catch, and abundance also measured as part of my work. Weekly survey were taken, and other water quality parameters, that is physical and chemical parameters, also checked for studies. The taxonomic identification were done based on the this is one of our project related with the venom proteins of jellyfishes. So we have to isolate the venom using liquid chromatography and mass spectrometry. We can identify the protein from the nematocyst cell. These are the results. We have 54 species of jellyfishes, including 48 Skyvosova and 8 Cubosova recorded previously from India. In Sova and 2 Cubosova from our coast. Among this, 6 species are causing severe blooms during some season, especially after monsoon in our coast. Sonorisa malensis, Netrostomus irrescens, Cyanosaki, and Acromitis flagellatus are the major bloomers in our coast. The protein derived from the nematocyst show close relation with some of the venomous snake, especially the pit viper and the cobras. These are the images showing the blooms in different seasons. In this Acromitis flagellatus, Kerala. We are check the environmental parameters to know the reason behind jellyfish. We got a good correlation between environmental parameters. That means the sea surface temperature have a positive correlation with jellyfish. When the temperature is increasing, jellyfish population also increasing. The dissolved oxygen. Dissolved oxygen is another important parameters for jellyfish bloom. Jellyfishes can survive in hypoxic water. 1 ppm or 1 mg per liter oxygen condition, it can survive. So hypoxic condition also helpful for jellyfish. pH doesn't have much role on proliferation or any other dynamics of jellyfish bloom. The salinity also have a positive relation with the jellyfish. When the salinity increases, the jellyfish population also increases. The zooplankton, especially the copypods are the preferred food items of jellyfish. When the copypod is abundant in the water, the jellyfish also abundant in the water. In addition to that, some coastal activities, constructions, seawall, also helpful for the attachment of jellyfish polyp to that area. In addition to that, the eutrophication, which imparts several dissolved organic material to the water, which lead to the primary productivity, that is especially the chlorophyll A productivity, which enhance zooplanktons. This condition will help for jellyfish. In addition to that, overfishing also a major reason for jellyfish. When we catch all the topmost predators, the predators of jellyfish will be less. That will help to raise jellyfish in our areas. These are my publication. I was published 2 papers in the 6th International Jellyfish Bloom Symposium in the last 2019. I got 2 opportunity for a training in Smithsonian Tropical Institute and for VOYO in this year. But unfortunately, due to the corona outbreak, this program is postponed to next year. I'm recommending something about some work for after COVID. We can do the field work with the support of fishermen. We can say our workable language to the fishermen. If they know this work is beneficial for them, they will help us without any remunerations. Modern molecular techniques. We collected some specimens and preserved in ethanol for a molecular analysis. We can do more and more molecular work in this institution, and my collaboration with several institutions. This will help to reduce the cost and to improve and better improve our research work and hello.
Thank you, Mr. Riaz. And
Okay.
this is open for discussion. Any specific questions to Riaz? Yeah, Dr. Dinesh.
Yes, thanks for the excellent presentation.
Thank you.
Yeah, so you mentioned about the temperature influence on its proliferation rate, right?
Yeah, yeah.
So when you count or when you look at the population density, so what time exactly you found that dense population? Exactly in the temperature time, or it's after the temperature time?
One thing that, for example, I am saying about one Escherichia species, that is Acromatus flagellatus, which is the number of jellyfishes is more during summer season. We studied about the life cycle of that species. We got the polyps and all the life stages in summer time. That is February, March time, and the peak, the proliferation, the jellyfishes bloom and is peak at May.
Okay.
Okay.
April, May month.
Yeah.
Temperature also increasing the proliferation of jellyfish.
Dr. Sarangi.
Yes, sir. Actually, good work. Jellyfish is an indicator, or another and few queries. What is the commercial importance of jellyfish? And third one, what is the relationship with fishery? Hello.
Yes.
Yeah. Could you?
Network issue.
Hello.
Ah.
Yeah. Is it an indicator? Yeah, jellyfish is an indicator. And other query. Hello.
Yes, sir.
Yeah.
Yeah,
and
yeah.
another, yes, other query is that what is the economical commercial importance, and third one is what is the relationship with fishery.
Okay, I'm answering one by one. It's we can consider it's an indicator because in our study the eutrophication is also influencing for jellyfish bloom. When the increase in the phosphate, silicate and phosphate, and chlorophyll A concentration also improve jellyfish population also. In the fishery, how it is impacted the fishery, because the main feeds of jellyfishes is the copepod and the osteoplankton, primarily the larvae of fishes and the eggs. Some of the studies from the CMFR indicated that the oil sardine, the population of oil sardine is declined due to the population, increase in population jellyfish, because the jellyfishes are eating more and more. So it's one of the important issue, declining fishery populations, fish populations.
Thank
Another
you.
One of the thing is
okay, thank you. Yeah.
Thank you, Riaz. We have actually crossed the time barrier.
Yeah, time.
And then we'll hear quickly from the panelists. I'll start with Baban Ingole, Dr. Baban Ingole, sir. On the generally on the presentations, and then how will we move ahead? Yeah.
Yeah, thank you very much. Excellent program. Overall, all the presentations were very good, very informative. Some of the were of very high standard. There is no doubt about it. I have enjoyed most of these presentation because some of them were the new ideas, and I compliment all the presenters for the excellent presentation. My take-home message is one. I wanted to say something with respect to the last presentation, and it is applicable to others also, because there are majority on the coral reef. Last was on jellyfish, and this is to Dr. Biju Kumar also. I thought maybe taking some help from Dr. Sarangi, will it be possible to come out with some sort of a correlation of jellyfish blooming and the environmental parameters? If we can, come out with some, data that will be very helpful for the fishery management. I think so. It will, maybe possible, but we'll have to work for that. So this is just a suggestion.
Yeah,
I thought about it. Second one was about 2-3 choral presentations, and it was basically for the transplantation. I thought we have good people working around, and good techniques they will be providing in future for the coral restorations, and such projects needs to be encouraged. There was one more presentation where I thought I can say something about it, and that was about the seabirds. And because that is another study, very limited knowledge we have got it, but they have done an excellent work on using the passenger ships and those, because there are limited facilities available for such studies. But let me just tell them that if they can come out with a good proposal, probably Government of India, FAO, and the International Tuna Fish Union, they will have the funding. Particularly with the Government of India, where I am working right now with MOES, they may have some funding, because there is a need for such data under the BBNJ program, Biodiversity Beyond the National Jurisdiction. And some of the area, for example, India is interested from the deep sea mining. There is no data available about the seabirds in the deep sea, as well as cetaceans. Therefore, if somebody can come out with the proposal, particularly this group, because they have good techniques, and if they can come out, there could be some funding available in the future. That's what I wanted to say. But as far as coming out with some special issues or something, there are very good publication, very good presentations. Original data has been presented, and I certainly look forward for some good publication from this. Thank you once again.
Thank you, thank you, Dr. Baban Igole, for your time. And we'll hear from Sanjay. Sanjay, are you around? Sanjay Molur.
Yes, I'm here.
Yeah.
I've been listening to all the presentations. I think done an excellent job of the work conducted in the field, plus the presentations put together. I think it's congratulations to everyone. I really enjoyed all the presentations and learned a lot. So my take on this, a couple of them, is that I think in one of the very first talks, it was emphasized that it would be better if everybody studying in the marine world comes together and understands what each other is doing, so that resources can be consolidated, and perhaps more taxonomic studies can be done if people know what the other is doing, and know who's doing what and what is required from a certain area. And from that perspective, a couple of things. One is that perhaps it would be good to have a forum wherein all the marine experts learn about all the other groups in terms of in a workshop, so that the basics is understood, and that they can complement each other's studies and work.
Yeah, and also, Sanjay, there is one issue with regard to India, is that we are very poor in undertaking multi-taxa inventory.
Exactly.
It's actually waste of money. A group going in the ocean and doing a survey on a specific group, and if it is a multi-taxa inventory, that will also save the money.
Absolutely. No, that's exactly my point. So if one is that, yes, I mean, we're all in our own little wells. I mean, it's not to point fingers here, but in the sense that in any field across wildlife research, it's the same thing. If I'm studying amphibians, I would not look at other organisms. I mean, it's as simple as that. I think in this field, it is even more relevant because there are not many people working in this in this domain. And so it makes more sense if multi-species studies can be incorporated. Of course, not everybody can be in those expeditions. So if the person who's responsible in going on an expedition can perhaps do certain basic studies or basic collections for others, then everybody will be benefited. That's one thing. The other thing is to perhaps on the same lines have a coordinating group. I would say Dr. Biju Kumar is perfectly placed in perhaps coordinating everybody who's working on marine research to help facilitate this kind of a collaborative undertaking. So, and if everybody puts in that information, pulls in what they're doing, and keeps that information current, then everybody benefits. So, I mean, these were just two of the thoughts that occurred to me. I mean, there are many more that we can talk about, but I don't want to hog this entire discussion. So I think I'll move on from here, just to emphasize again those 2 points. One being the network, the other being the training. I think these two will take marine research in India a long way.
Thank you, Sanjay and Dr. Rajiv.
So I think from parasites to birds, we've been treated to some really fascinating talks by all these young researchers. So I'm not an expert on these taxa to comment on the the quality of these presentations, but there's some general messages that came out from all these talks. One was that collaboration is the key, and I think this is a time where collaboration and networking is the most important, among all the different time periods we had, because without linkages, networking, and collaborations, I don't think research is going to move ahead, at least in the next month or few years. So that is the first point. And the second point is, as taxonomists, we've been always visiting museums, repositories throughout the world in different places in India. So all this is going to come to a standstill, at least for the next few months and years. Although we've been always talking about digitization of museum specimens and all that, so nothing has happened so far in India. And this would have been one of the best times to make use of digitized museum specimens. And I think the world will definitely face challenges like this in the future. And if we start thinking about digitizing our museum collections, digitizing our marine collections, not only marine, any for that matter, any museum collections, I think in the future we would be in a better position. And then there are also people who talked about citizen science. I think that's again the future for collecting data, for information gathering, especially again with this travel bans and things in place. So involving public, the common public in research is important in the future. And then, like what Sanjay said, it's very important that we maximize sampling efforts, maximize funding, and get the most out of even small field surveys. So whoever has a chance to go to the field and collect samples, I think the entire effort should be maximized so that many people would benefit, and it's just not that group or that person is benefiting. And I think one of the things that has not been talked about in this group is social science-based research, where people have to deal with communities, have to deal with people, respondents. And I think things like questionnaire surveys and so on. So those are again issues that we need to consider, and I'm sure there are people who could be doing that. So I think with that, I think this was a very fantastic experience, sharing knowledge, hearing all these people what they're doing, and how we all as a collective group can get together and face the challenges of COVID and other sort of issues in the future. So thank you, Dr. Biju, for inviting me to be an observer and giving me an opportunity to hear these great presentations.
Thank you, Dr. Rajiv, and over to you, Deepak.
Thank you all. And so first I would just like to answer the question Dr. Rafi asked, how from a marine biologist, how I became an integral coastal management planner now. Taxonomy is my passion, Dr. Rafi, and it's in the heart always. Most of my colleagues and my senior colleagues know that I spend so my time is now very little to work on, but I'm happy to say that I was able to prepare an army of young researchers to whom I can delegate the work and supervise them. Right, that's an advantage I have. And of course, livelihood, we are multitasking now. And the ministry, they ask you to change your profession, from taxonomy, jump to ICDM, I don't know tomorrow what I'm going to be. Right, but it's all experience is what I would say. And I would just like to encourage the youngsters and say that, see, what is passionate to your heart, please go ahead with that. But when it comes to career, you can be a taxonomist yourself, but then if your office job demands you to do something else, it's an additional advantage. You can still grow in that. So we can keep parallelly growing together. So that was a response to what Rafi wanted me to answer. And as all the senior panelists spoke, they've covered almost everything. My only apprehension is that we're talking about now Sagarmala, so development along the entire coast of India. And we are going to talk about blue economy next. Already India started to work on that. So we should be also very careful on how conservation can gel along well with development, right? The ministries, all the ministries are now into development along the coast. More ports coming up. And when the presentation was Al-Battush, when he presented, Lakshadweep. Now turtles are nesting there, but where are they nesting now? Again, is going to be a question because now the administration wants more of shoreline protection because the island is eroding. So it's a very thin, rope that we are, thin thread that we are in actually. All said and done, I think capacity building also is very important. I'm very happy in the last 3 years we have seen a lot of training programs that have happened, where people who are in mid taxonomic research, people who are in fresh, starting to research, they had an opportunity to have a feel of international and national experts. So that's also another point, and very neatly put by Dr. Rajiv, the social part, because I think we have to rope in the community into everything. Any survey, I think the indigenous traditional knowledge that they have, something very important. That is again a good eye-opener for us, especially research, because if we don't gel the local knowledge and our experience together, then I don't think science will go any further. I thank Dr. Biju for inviting me to be as an observer, and it's a real privilege to meet old friends, through this particular webinar. Thank you so much.
Thank you, Deepak. Dr. Sarangi, a quick response.
Yeah, excellent platform to discuss with youngsters and getting their views, ideas, and the future program projects they are going to handle post-COVID era scenario, and great guidance by this panelist, and it's a great successful program, I can say, and on this World Ocean Day. So I hope all these youngsters, young breeds of researchers, they able to do a lot of great with enthusiasm, able to great activity they should able to handle for this national progress in achieving the level of marine research to sky high. And India of course is doing great, like NIO and NIOT, all Cochin University, then Kerala University, and several research institutes are there. So, but still, I feel that many frontiers are new, and I saw good works like many conservation work activities, and works like seabirds, then turtles, and then seahorses, and several new, in addition to coral, the new activities has been discussed in this forum. That one malacologist, mollusk, also marine mollusk, one guy discussed, researcher discussed. So that's why I felt it's a lot of scope is there to explore new researches, new frontiers in marine activities, and they should able to have the young researchers. My opinion, advice, and request there, they should go through thoroughly the literatures. Literatures are available, because earlier days it is not there through books, library, we had to go through. now globally means connected, world is connected, so across, so they have to go through all these research means literatures, even for decades, means many decades, centuries, it is available, so they should go through, and I feel they should try to enhance this research atmosphere arena of Indian means marine scientific, whatever the compendium is there for all subjects. They should able to means explore and try to bring the means Indian marine research to sky high. That is my request and suggestion. Hope all will achieve to this kind of target to have their research publications in very high quality journals and high impact journals. All the best. Thank you.
Thank you, Dr. Sarangi. And at the end of the day, end of the Ocean Day, just like the theme says, innovation for sustainable ocean. We discussed some of the innovative ideas, and then this is a time to carry it forward. Just like what Dr. Rajiv said, the taxonomies also have to grow vertically and laterally in the real sense, because we need to have more transdisciplinary and interdisciplinary work, including with the social sciences, so that we can at least influence the policymakers and showcase the value of taxonomy and biodiversity documentation. Hope this will be an initiative in that direction, and we can move ahead with that positive note. And I request Dr. Suvarna Devi to propose the vote of thanks.
Okay, good evening all. So we have reached the end of our webinar, Voice of Youth for Ocean. So on behalf of Aquatic Biology and Fisheries, I thank all the participants watching the webinar. And I wish special thanks are due to Dr. Baban Ningole, who is a full-time visiting scientist of National Center for Polar and Ocean Research, Goa, and also the former head and chief scientist, NIO, Goa. I wish to express sincere gratitude to Dr. Sanjay Molur, who is the executive director and trustee of Zoo Outreach Organization and IUCN Expert Committee, and publishers of Zooprint and chief editor of JOT. My sincere thanks are due to Dipani Sutaria, who is a freelance researcher working in the field of conservation, biology, and behavioral ecology. My immense gratitude to Dr. Deepak Samuel, scientist in CMR, Conservation of Coastal and Marine Resource Division, from Ministry of Environment, Forest and Climate Change, Government of India, and sincere gratitude to Dr. Rajiv Raghavan, Assistant Professor from KUFOS, who is a coordinator of IUCN SSA Freshwater Fish Red List Authority, Asia. Special thanks are also due to for young scientists and researchers, Dr. Prakash, Reshma Dilip, Dr. Jabir, Dr. Tinesh, Dr. Ravichandra, Dr. Sarangi, Dr. Ravinish, Dr. Chandran, Dr. Anish, Ramvilas, Shalu, Albadush, and Riaz. So we hope that all those who have listened this webinar will definitely take away interesting information regarding the biodiversity of our ocean. The topic we have dealt today is vast, that is from microbes to coral reefs to seabirds. So on behalf of Aquatic Biology and Fisheries, and thank each and every one of the participating, those who have participated in this webinar, thank you for your patience. I wish you all a very good evening. Thank you.
Thank you. Thank you all. Good night to all.
Yeah, thank you all. Good night.
Good night.
Good night.
Good night. Thank you.
Thank you.